Hello everyone. So today we'll learn how to install MATLAB and COBRA Toolbox on your Windows computer, and later on we will learn flux balance analysis and of genome scale model using the MATLAB and COBRA Toolbox. So let's see how to install MATLAB. So we need to download the MATLAB trial version. We go to the website mathworks.com, and here we can sign in or create an account using which we can install a MATLAB file. So if you don't have an account, we can create one by just filling in uh, all the details here. You can create an account, or since I have an already have an account with this email ID, I can just login using my email ID and password. Yeah. So using this icon, we can install MATLAB. Download for Windows. We need to open this file. Yeah, click on the this trial license. Set the default location. So here we need to check only the basic MATLAB on basic MATLAB. We don't have to select any other option. It is just MATLAB that we require for running the Cobra toolbox. Yeah, add shortcut to desktop. So while the MATLAB is being installed, we'll see how to install Cobra toolbox. So that to install Cobra toolbox, uh, we need to go to the website of Cobra opencobra.github. We can search using. So we can go directly on the installation page. Here we see what are the system requirements for installing the Cobra toolbox. The first requirement is itself the MATLAB. Second is you need to install Git. You can check if you have a working installation of Git by typing in the terminal or in the command prompt. In case of Windows, we can type command in the command prompt like this command git hash hash version, and it will show you an output if it is installed properly. Similarly, you need to install curl, and you can check by typing in this command in the command prompt in the terminal or in the command prompt in case of Windows. So let's check if we have a working installation of Git. I just copy this command. Now I'll open my command prompt. And I will paste this command here and see. So it is saying that it is not recognized as an internal or external command. So it is not installed on my system. I need to install it. Similarly, for curl, I will check by copying this command and pasting it into the command prompt. Yes. So I have a working installation of curl on my system. I need not to install it again. Okay. So we need to install Git. Let's see. So it is, yeah, it is about to complete. Okay. Okay. So you can see there here if we don't have a working installation of Git, Mac users can install Git using uh, this export command line tools. Windows users need to go to this website, and from there we can install Git on our system. Yes. So since I have a 64-bit Windows machine, I need to download this setup file. Open it. Yes. Next. Next. Yes. Next. 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 Let's get decide. Here you need to select the third option. Next. 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 Here you need to select the second option. Next. 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 Install. MATLAB is installed properly. So we need to launch git bash. So this is the git bash window, and we will be using it to install the Cobra Toolbox. So to download and install the Cobra Toolbox, to download the repository, the folder will be of Cobra Toolbox will be created. You can clone the repository using this command. So I will be copying this command, copy, and here is my git bash. This is the git bash, and right click, paste, and press enter. So it is now copying all the files into the folder Cobra Toolbox. Yeah, so the Cobra Toolbox has been downloaded. We need to exit here. Now we will okay, exit. Now we will open our MATLAB and see if it is working. So the Cobra Toolbox folder has been created here by Git, and all the files have been downloaded here. This is my documents folder. Here, going backwards, and I select the administrative user, and here you can find the downloaded Cobra Toolbox folder. We will need this later while setting the path in the MATLAB. So to initialize the Cobra Toolbox, we need to enter the command init Cobra Toolbox false. So we are getting an error here because we have not set we have not set in the path for Cobra Toolbox in the MATLAB. So we need to put it into the path. So to do that, just go to this option, set path. Here, select this option, add with subfolders, and here we need to select the our folder, the downloaded folder of Cobra Toolbox. We need to go to the administrator user, use, select this Cobra Toolbox folder, downloaded here, select folder. So all the files have been copied here. We need to click on save and then close. Now if we enter the same command and we will try to initialize the Cobra Toolbox. Yes, so it is properly initialized.
Yes, check the chat. Yeah, there are some messages in the chat. Okay. Can you all hear me well? Yes. All the participants, all participants can hear me well. Okay, thank you. So some of the online participants did not join the earlier hands-on on the MATLAB. So I will cover the basic MATLAB basics again for some time. So as I'll share my computer screen. Right? So the online participants can see my screen, the MATLAB screen.
Yes, thank you.
Okay. Okay. So this is the MATLAB home screen. Give you some basic idea. The central panel is called as the command window, where we enter our commands, and this leftmost panel is called as the current folder or working directory. So in this panel, we can access our files into the MATLAB program, into the MATLAB basically. We can select any folder here in the current folder, and so I'll select another folder here. So if I select this, for example, this folder, all the files which are there in the folder will be shown here. So maybe there are no MATLAB files in this folder. I need to download them. I'll quickly download the MATLAB files. Just wait for a minute. So I'll change my current folder here from here, and I'll go to the desktop where I have saved some files. So you can see some files appear here, and I can now access all these files into the MATLAB. The rightmost panel is called as the workspace. As you can see some of the data here. So in the workspace, all the data that we create or variables that we generate, they are saved into the MATLAB memory, and they are displayed here into the workspace. As we have some data generated in the previous session, we can clear the data in the memory of MATLAB using clear command, and you can see the data being erased from the workspace. So through entering CLC, we can clear the command window. So we'll try to generate some data. For example, we can try, I will be generating a data named A and give it a value 1. So you can see that I have created a data, a variable named A, and it is having a value 1 saved in it. Similarly, I can create another variable, say I will name it on my name and give it a value of 25. So another variable is generated in my name and it is having a value 25. Okay. So basically, we can also perform numerical calculations, numerical operations through this using these variables. So if I want to add these two variables, I can simply enter A plus Piyush, and the answer comes out as 26. So another variable is generated in the name of answer, and the value is 26. So we can perform any numerical operations using these variables. So now we can clear it. These are some basic examples, and other types of data can also be generated and processed, as we will see later in the session. MATLAB can also we can also import large data files using this import data button, and we can also analyze those data using the various commands, and we can analyze our data and produce various results using the different commands that are available in the MATLAB. So since our topic of today's hands-on is flux balance analysis using the COBRA toolbox. So we'll try to do data analysis using the genome scale metabolic models. So to start with, the COBRA toolbox had to be loaded into the MATLAB working environment, and to load the COBRA toolbox, use a command called as init COBRA toolbox. We enter false, as we don't want to update this COBRA toolbox. And you can see that the packages are being loaded into the memory of MATLAB. Okay, so the COBRA toolbox is loaded into the MATLAB. As we can see, this command prompt is being again displayed. So we are ready to type in new commands. So the first thing that we have to do is we have to load our model, our genome scale model, or any COBRA model into the MATLAB, so that we can use that model to work. For example, you can see here into the current folder, there are various files. Okay. So yeah, there is we need basically the genome scale models. They are saved into systems SBML. They are saved as SBML file formats, and we have to download these SBML or .xml files. We can download these SBML or XML files from the internet. There are hundreds of genome scale models. They are available at the UCSD websites. People have generated these models, and they have submitted these SBML files onto the database of UCSD. So I'll try to show you how you can download these genome scale metabolic models. You just need to go to the website. I think I need to change my settings to share my current screen so that people online to follow. Firstly, I should select this first one. Oh. So we will go to the UCSD website. I need to put some more details. So yeah, the correct website is bigg.ucsd.edu. You can write down this website on this URL. So this is the database of the genome scale metabolic models. So if I want to search for E. coli, yes, you can see different metabolic models. These are basically genome scale metabolic models. The first model is E. coli core. So this E. coli core model is basically a short model which is here, which they have made for learning purpose, so that because it runs very quickly, it loads very quickly, it has basically limited number of genes. So you can analyze this model very quickly. These models, IEC, these are models, basically these are genome scale models. You can see here the details about these models. The first, the E. coli core model, have 72 metabolites and around 100 genes and 95 reactions. These are the genome scale models. Basically, all of the genes that are present in the organisms, they have included. You can see different strains here. So today we will be working with IJO1366 model. We can choose any model to analyze, and I will choose IJO1366. Can I see it here? IJO1366 is not here.
From the bottom, I think.
You can put it in the search database, you will get it.
Yeah. So this is the ID of the model, IJO1366, and the organism is, as you are able to see, it is E. coli K12 MG1655 strain. Okay. So the number of genes are 1,367, around 2,500 reactions, and around 1,800 metabolites are included in this model. Now, how to download? You will click on this model ID, maybe, or something is there. Yeah. So, yeah. So this is the SBML or .xml file. We will just download this file. The file is downloaded. Another version that we can use in MATLAB, this is a .mat file, which is a MATLAB format of the file of the model. This file we can also use while working in the model, in the MATLAB. Sorry. The .xml file can be used with other softwares also, like CobraPy, which is a Python-based package. So I have downloaded the files, and now to access these files into the Cobra Toolbox, I have to transfer these downloaded files into my working directory. Since my working directory is there on desktop, copy these two files and paste it into my working directory. Okay, this file was already here.
So working directory would be any new folder we can make, or is it we have to make for something like that?
Can you speak a bit louder, ma'am?
I'm saying that you are saying now that this file will go in the working directory.
Yes.
So working directory is like any folder that we are making, or is where we have to specify?
Working directory can be any folder where you save your files, basically.
It doesn't have to be in that where we have.
Yeah, you can select here because working directory you select here. You can browse any folder where your data, where your models are saved. Okay. So this was my working directory. So I'll clear my command window using this CLC command. Now, to load the XML version or the SBML version of the genome scale model, I will use the command and I will save the model as a variable named model ORI for original. As we will be making some changes while doing the FBA, flux balance analysis, and we will be changing the model according to our requirements. So I want to first save the model with a different variable so that we can reload the original model whenever we want. Okay. So the command to load the model is read CB model. CB stands for COBRA. Since all these models are COBRA models. And here I have to specify the name of the file, and the name of the file is IJO1366.xml. All you people are doing it on your laptops?
Is it possible to zoom it a little?
Okay. I think
Copy and paste it in PowerPoint.
okay, copy and paste. Can we do it from here? No. One second, ma'am. I'll figure it out.
There is command.
Sorry? Yeah. You have that booklet, right? You are having that booklet, and you can use that booklet. Can I have that booklet also so that I can specify the page number?
21.
I will tell. Okay.
Page number 21.
Why it is taking too much time here? So all of you have loaded the model into your MATLAB environment. It's loading. The online participants, are you able to see that the model file has been loaded into the MATLAB?
Yes. Thank you.
And there is a variable and it has been saved as the name model_ORI.
It's loaded.
In the booklet, _ORI, it is not written. It's
Okay.
written only model.
It is written only model in the booklet. So it doesn't matter basically. It is a name, so we can name it anyway. It is a variable.
Just put don't put the directly.
So meanwhile, you can see the online participants can see that some of the details are being displayed into the command window. This structure stands for data type. Basically, this model this model file is a type of data, and MATLAB saves this model file into a type of data which is called as a structure. Because this model file is having different data in itself, and as you can see that there are metabolites, lower bound reactions, lower bounds, upper bounds. I will tell you what these things are: genes, rules, gene names, compound names. Okay, so all these things are saved or included into the model file, which has been which is the MATLAB has read. Sorry, the COBRA Toolbox has read using the command read series. So, are you able to load also, people? All of you? You're getting an error message. Can you help her out? I will be very happy if you can help your colleagues.
No, it is not. That is the one.
No,
I think you are making mistake.
for loading this one.
It is not.
Keep this file in this MATLAB
It is not
library. Directly, otherwise
You just copy paste and make it. And it's capital. Yeah, just make it. Have you downloaded the general variant was downloaded here? Select. Okay, select. Select. Now you
So now we'll try to see what kind of data is included into this model file. We can click here on the name of this variable, and you will see that all the data included fields are being displayed, and we can see the details here. So the S, this field includes the stoichiometric matrix that the Dr. Raghunathan was describing about in your first lecture today. You can see the same matrix with rows and columns, and this matrix is basically, it includes all the reactions and it is generated by all the metabolites which are being used in all the reactions. The rows, these rows represents basically the metabolites, the number of metabolites that are included into the model, and the columns represent the fluxes, the reactions which these metabolites are being used into the metabolic network of this organism. So we can see that this metabolite first is not being used in this first reactions, since the entry here is 0. It's not, neither it is being used or not, it is not produced. Okay, if it is being used here, it would have been written minus some number. For example, one molecule is being used, then it would have been written -1, or 2, then it would have been written -2. So it is basically the stoichiometry. It represents the stoichiometry of the metabolite into the network. So other things, these are the list of the metabolites. So you can see here different metabolite names of the metabolites. Basically, these are the identifiers of the metabolites, not the proper names. Identifier is the name basically in which the MATLAB understands the metabolite or the reaction. We write down the reactions into the model, the way in which we enter the name of the metabolite into the model. You can see that there is a small c written over here. Sorry?
Alexandra.
Someone is raising some hand. Okay. Yeah. How to see?
Alexandra is.
Yeah, may I just ask something? Is there somewhere a list where we can know these identifiers, how they are related to the actual name of the compound?
Yeah, ma'am. We can see. I will tell you. So I'll explain. So these are the identifiers of the metabolites, and there will be names also. There will be another.
Okay.
Another list also. So.
Okay. Thank you.
This one. This is the list which explains the name, which is for the names of the metabolites. You can see here.
Okay, and the numbers are the same.
Numbers, yeah.
Okay.
Yeah. Serial numbers are same.
Yeah. Okay. Thank you. Sorry.
Okay. Welcome. All the questions are welcome. If anybody is having any questions, they are welcome to ask. Okay. Since it is most of you are doing it for the first time, you will be having many doubts. And I request you to clarify your doubts the time you have at the same time, because you will be forgetting later, or you can note down it if you want to ask later, and you can ask it anytime. So other lists are also here. Genes are here. This is the list of genes. As in previous talks, you have heard that the genes are in E. coli are named like this B numbers. So here is also the same B numbers you can see. Okay. So these things, we call these different lists as the attributes of the model. So these are the different attributes of the models. Reactions. So these are the different reactions that are included.
Needed to allow.
You will be seeing, you can see these two attributes called as LB and UB. LB stands for lower bound and UB stands for upper bound. So lower bound is the minimum value, which it stands for the lower bound represents the minimum flux that is allowed for a particular reaction that has been given into, that we have preset it, prefixed into the model. And the upper bound is the value which we have predefined, that is a maximum value the flux can take, any reaction can take. Okay, so we can change these lower bounds and upper bounds according to our requirement, and we will see how the model behaves. Okay, and we will calculate we will try to fix, we will try to change some of the bounds of the reactions, and we will see how the fluxes of other reactions will change. For example, the growth rate of the model will change. It's time for lunch. Okay, so we will stop here and we will restart up at 2. We are stopping here for lunch. We will restart again from here only at 2. Okay, thank you. I request online participants to join us at 2 p.m. Indian Standard Time, IST. Thank you.
Ask them to write the comment if they have any doubt in the charts.
They can speak up anytime.
Even
They
if
can.
they have, And please don't sign out, okay? Yeah.
Okay, I will not sign out. I will be, this window will be present like that only. You people also can stay logged in.
You can stop sharing and maybe turn off our camera, but camera and mic, but you can let them.
Okay.
Oh, 2017. Okay, it was by 5 years of BTech and 15 started. 2028. How come?
Okay. Hello, everyone. Are you able to see the MATLAB screen?
Yeah, thank you.
So now we shall proceed from where we left. As you are already aware that we uploaded the model into the variable named model_ORI, so that the original model is saved. We can later on reload the original model. So now for the simplicity, we will again define a new variable model, and we will save it. We will give equal to the original model. So as you can see, the same model, the model variable is now equal to the, it is the same as the original model. So all the changes that we will do with the variable model, and our original model will remain unaffected. And whenever we will need the original model, we will again equate the model variable model with the model_ORI, and the original model will be restored. So we will test some of the basic commands in the COBRA toolbox. The offline participants can refer to their booklets and just try to type in the commands. Yeah, on the page number 22, the second command. So this command is called as write CB model, write COBRA model. The commands have to be typed exactly the way they are written, because these are basically functions that are predefined in the COBRA toolbox. So we cannot, if we type the name like CB, where I have not capitalized the letter C, okay, so nothing will happen. It will give an error. As you can see, so the MATLAB is really good. It is also suggesting the right correct command. Did you mean write CB model? The correct command is capital C with capital M. Okay, so what is the function of this command? Using this function, we can write the XML file or the SBML file of the model into a XLS file or an Excel sheet. So all the data that is there in the SBML file, we can transport or export the data into an Excel sheet. So since we can easily read all the data that is saved in the SBML file, and we can easily read it into an Excel sheet. So the different parameters that need to be entered with the command are model. Model is the XML file or the structure in the MATLAB that we want to export. XLS stands for the format into which we want to export our file, and here this is the name that we want to give that file. So since the model is named as IGO1366, we will use the same name. The names of the model have to be put with the single quotes, and the format is also has also been true with the entered with the single quotes. So now, yeah,
Yeah, it asking model requires Simulink.
I cannot see the question.
No, here.
Yeah, the computer is screen. Hey, your path should be selected.
Yeah, I wrote write CV model and all things. So it writing model requires Simulink.
Model requires, have you defined the, I have defined another variable, see, model. And I have equated it with the original model,
Yeah.
original variable, yeah. So you have to define another name. Not here, type in another variable. You have to define another variable named model and equate it with the model underscore ORI.
Okay.
Okay.
First it was saved with model underscore core AI. Now it is set it to the model.
Model.
Model is equal to model.
Yeah.
Score over.
And model. We originally imported the as given file as the name model underscore ORI. Now we have defined another variable
Simulink.
model.
We have to write model equal to model
Underscore ORI.
If you put in reverse,
No, you will, if you put in reverse, it will not work.
yeah, it's again saying the Simulink. Okay.
Yeah.
Right, CV model.
So you can see that in the current directory, another file has been created, IGO1366.xls. Okay, so I will go to this directory and see what has been written into the,
Okay.
what is in this file.
Bye-bye.
Here is that file. This is the IGO1366 file. No, this is not the file. This is, I downloaded.
Same, that is the one. You can see the time
Then
when it
I'll-
was placed. Refresh it. Auto modified RMs.
No.
Score model.
Go to
You
the
can directly click on that MATLAB window.
data modified.
Name के
Data modified.
Okay.
The recent one. Anything over there and that.
No, it is, it was downloaded around 147. It is not the new file.
So for us.
directly click on that MATLAB.
Yeah. You are able to create this file?
Yes.
Okay.
Refresh कर लो.
Yeah, I need to refresh it. For the online participants, I have to show it, right?
Yeah, I got the Excel
I will reopen
file
this directory.
name core model Excel. Okay.
Light, come light नहीं है.
But it is still there.
Yeah, I got the Excel file.
You will, here it is not like that.
You can open it from here, right? Just MATLAB के विंडो से ही ओपन कर सकते हैं वहाँ पे डबल क्लिक करके.
Yeah, वहाँ पे भी download it. एक बार rerun करो.
Okay, I will try to rerun this file. Rerun this command. And I will give another name, okay, so that we can see that the file has been created just now. Yeah. The online participants you can see that a new file has been generated, IGO1366_new, and it is a Excel file, as you can see from the format .xls, and we will try to see the data.
Change कर देना. Directly change कर देना.
यहाँ पे जो फाइल नेम अपन को देना है वो जो ऑर्गेनिज्म का अपन ने लिया उसका नाम देना है, जैसे इसमें कोर मॉडल लिखा है
कहाँ पर?
प्लेट में दे रखा है कोर मॉडल।
ये तो नाम है, it is only name. So we can give any name, whatever we like to give.
Okay, it will take that data only.
Yeah, as you can see right now, I've given a different name, right?
Okay. It's fine.
So,
Right. Yeah,
Yeah, I got this.
yeah. So I will just open it. So
Yeah.
all of you are able to see.
Yes.
Yes, yes.
So it is now very easy for us to see what kind of reactions, reaction names, what are the lower bounds, what are the upper bounds, and whether it is set as an objective function or not. I will tell later what are the different things. So as you can see that this reaction, and what is the description? It is a chloramphenicol exchange reaction, and this is showing the reaction structure. Yeah, this EX means exchange. So exchange reactions are those reactions in which a metabolite is being exchanged from the cytosol to the external environment. Okay. We will be using these exchange reactions when we will be simulating our model for, and we will try, we will be adding butanol pathway into the model. So you can see all these EX reactions. So these are all reactions which are trying to exchange these metabolites from the cell to the environment. So this is a very easy for us here, so we can to see what is there in the model. So as you can see that here the at lower bound 0 is there and upper bound is 1000. So what is the meaning of this lower and upper bound? As I told you earlier, that the 0 means that this reaction, it can have a minimum value of 0. It can have a minimum flux value of 0, and the upper bound means that it can have a maximum allowed value of 1000. It cannot go above 1000. So why we have choose 1000? Because it is an arbitrarily, we choose an arbitrarily large value, so we have choose it as 1000. Okay. And here, this is a good example to show. This is an exchange reaction for glucose. Okay, as it is written here, EXGLC, and the description is given as D-glucose exchange. So with the help of this reaction, we can the cell can take up. We can give the cell glucose, and the cell can take up glucose from the environment. Okay, and the lower bound is set as -10. So the cell, as we have fixed the value of -10, so cell can maximally take lower bound as -10 millimoles per gram disaccharide per hour. The maximum flux that is allowed for glucose exchange from the environment is -10. This minus sign indicates that the glucose can be uptaken. It is an uptake rate. So the cell is taking from the environment. So the minus sign indicates that thing. Okay, and the maximum upper bound is 1000. Okay. So the cell can, it is not possible, the cell can, the positive value of upper bound means the cell can secrete a maximum glucose value of 1000. The maximum flux of the glucose exchange can be +1000. Plus sign means it will be secreting the glucose out of the cell into the environment, but it is not an practical, or it is not a practical thing. The cell will never secrete glucose. If it is secreting glucose, something is we are doing wrong. It will not be able to simulate. We will not be able to simulate the model when we will be giving a positive flux value. Okay. And another reaction I would like to introduce you is the oxygen exchange reaction, as we will be using it into our simulations. So yeah, it should be here. Leucine, lysine, malate exchange, mannose. Yeah, so this is here. Oxygen exchange, EXO2_E. So this reaction is oxygen exchange reaction. And you can see the lower bound is set as -1000, so that oxygen can be uptaken with a maximum value of 1000. The cell can take up oxygen from the environment with this flux. It is maximally allowed. It is not necessary that it will always take this much of oxygen, but it is allowed to take this much of oxygen for its growth. Okay, when we will try. So we will come back again. Okay, the second thing is the metabolite list here. So these are the different metabolites and their descriptions, their charge, and their KEGG IDs are also given here. And the compartment, that is whether the metabolite is present, where the metabolite is present. In case of E. coli, there is only one compartment, the cytosol. There are no any cell organelles present here. But in case of complex cells like eukaryotic cells, since there are different compartments, for example, in any algae, there will be compartments like chloroplast and mitochondria. Okay, so there we will divide the reactions and metabolites into the form of compartments also. Okay, here only one type of compartment will be present. So coming back to the model. Now we will try to run some simple commands. Okay, so how can we print the different different attributes according to our requirement? As you can see, the first command that is written here is genes. Yeah, you guys also type into your laptops.
One question, please.
Yeah.
We had lower bound and upper bound limit.
Yeah.
So the unit is moles or what?
Obviously, it is the bound of the reactions. So whatever the unit of reaction is.
Number of reaction or moles?
Number of reactions or moles?
Like you said that we have -10, so the glucose can taken up, will be taken up.
Yeah.
So so that the meaning of 10 is the unit or
a unit of flux.
Flux. Okay.
Yeah. So the command we will type is cell structure model dot genes bracket 1 to 10. Don't type the semicolon, as semicolon will prevent the printing of result into the command window. So don't type the semicolon in your windows. So what this command will do, it will print the first 10 genes that are added into the model, and it will print those 10 genes in the order first to 1 to 10. So you can see the result on my screen. So these are the first 10 genes that are there into the gene compartment, or sorry, the gene attribute that is there into the model. So this 1:10 specifies that we want the list of genes starting from the 1 to 10. If we will put here 10 to 20, it will print from the 10th gene to the 20th gene. Okay. So in this way, we can access the genes, the names of the genes that are there, or the gene IDs. Sorry, names of the genes. Okay. The next thing is gene ID. Gene ID means at what position a particular gene is there into the model. At what position? So type in the next command, gene IDs. If anyone is having any problem in understanding, if any online participant want to ask any question, please, you are welcome to ask at any point. You are welcome to interrupt me. So to identify the gene ID, the command is find gene IDs. Yes.
For example, here find gene IDs as well, right?
Right.
So the G is capital. And is it case sensitive?
Yeah. It is case sensitive
So
because it is a command.
okay, we have to give the exact.
Exact. Yeah, you have to give the exact command. Otherwise, it will show error. You can try to. You are welcome to figure it out.
Yeah.
Type in and see what happens.
So actually, good thing is here it will suggest like you may.
Right. This new feature has been added into the MATLAB, but it suggests the right command. So right now I have entered something wrong, so it has suggested, and automatically it has did my job. Right. So first 10 genes have the gene IDs 1, 2, 3, 4, 5, 6 as expected. In the serial wise, the gene IDs are the serial numbers basically. So this is the simple thing. But if we have any gene, suppose, and randomly we have got a name of a gene, suppose in your booklet it is given B0116, and we want to know at what position it is there, we will type in its name B0116. Okay, and see that it is there at 194th position. Okay. In the booklet, it is given 4 because the model that is there in the booklet, it is different. It is E.coli core model that has been used to simulate for the booklet. But here we have imported the different model, the E.coli Genome Scale model. Okay. So are we clear till yet? Okay. Any question from online participants? Okay, if I'm having any question, then I will get a notification here. Is it? Oh. So next thing that we can do is that we can print. We can also find out what reactions are associated with a certain gene. So the command for that is find reactions from gene. This command will help you to find out what reactions are associated with a particular gene, say this B0116 gene. So to print it out, we will write results, list result, find reactions from genes, model, gene identifier B0116. Okay, we can put in additional parameters also, but they are optional. The 0 and 1 are additional parameters. We do not need to enter them, but
What is this additional?
you can see them, but they are not necessary right now. They have some default values. They are not to be worried about. Okay, I'll try to remove them. I was having error in this command here also. Unrecognized field name GR.
मेरे पता चल गया ना लिखवा रहा
So yeah, the problem is here that
है
this command is saying that unrecognized field name GR roles. So the problem is that in this model, in this particular model, this field is not included. The modeler, the person who has made this model, he has not included this field into his model. That's why Cobra is not able to extract that information from the model. It is suggesting that unrecognized field name. Okay. So this problem can happen because different modelers can make their models differently, and sometimes they do not include all the fields. Okay, they just skip that. So you can we can try from the different models. For example, I will try to load the E.coli core model. You can also try it on your system. I will be, is it in there my working directory, the E.coli core model? It is not, but no worry. The MATLAB has the E.coli core model by default. So, okay, sorry, the COBRA toolbox. So we can type in the command load. E.coli core model. Okay, and we will give it a name, core model. Give it a name as core model and save it into the variable core model. Invalid expression. Yeah. So the problem is that I was trying to equate it. This problem was coming. We cannot use this load command directly with a reaction variable with a variable. So now you can see that this model file has been updated. This model variable has been updated. How do we know this? We can see that now the reactions are 95. Earlier we would have seen that there were more reactions, around 2000 reactions were there. So it is the core model that has been downloaded. Okay.
In my, it is showing that load requires model-based calibration toolbox.
Where, ma'am? Can you specify the line?
After I write load E.coli core model.mat, then it is coming. Load requires model-based calibration toolbox. After this, when I write this.
Samsha, can you check that she has typed the correctly?
.mat also लगा हाँ underscore दोनों के बाद underscore आगे भी underscore.
So any online participants are having MATLAB installed in their system?
मॉडल के आगे ये dot.
underscore, will be underscore.
Why it's showing invalid expression all the time?
No, because this load command cannot be used with this equals to sign. That's why it is showing that.
L is an small.
As you can see now, I just typed in model equals, and you can see the description is E. coli core model. This new model is E. coli core model.
How this one we are calling that. It is loaded.
Write the command load.
E.coli underscore core.
You set type the model. Model command. And model is equal to.
Okay, so Alexandra don't have installed. So no problem. You can see the you can see my computer, and we are recording this whole session.
Yeah.
We'll put it on YouTube. So whenever you install it, you can just check it out.
Yeah. That's the idea. Thank you very much.
And we can recheck what the model.ori model here earlier saved our genome scale model as model.ori_ori, and you can see here it is having a different model ID. Model ID is IGO1366. So this is the genome scale model.
आएगा load हो रहा है.
And this new model that I have done loaded was E. coli core model, and you can see the description here also. So E. coli core model is it comes by default with the Cobra Toolbox. When we install the Cobra Toolbox in our system, some files are there by default.
Can you show the command line again?
It will take 2 to 3 minutes.
Okay.
इसके पहले
Model. Before this.
without equals to which you have wrote. Yeah, this one.
Mat. Okay, I have to write mat.
Yeah, the .mat means it is a MATLAB file. Okay, so the MATLAB extension files can directly be loaded with the load command. Yeah, but for the HTML file, we have to use the command read CB model. Okay. Excuse me, ma'am. You are not able to follow. You are having a headache.
We didn't get this file.
Oh, you are doing it on your screen, right? With arrow and fine. So,
Didn't get this window.
right?
We didn't get this one.
You didn't understand this thing.
No, we didn't getting this.
You are not getting this thing.
Yeah.
Did you?
No, the previous one also. This one. Yeah, I wrote E. coli core model.mat.
Right.
And it doesn't show it.
You have typed, yeah. So model has been loaded there. Now type model.
It's not showing like the file.
Type model again.
Okay.
Type model again, right? Can you see the next command that I added?
Yeah, okay. Now it's
So now we will try again to find reaction from the gene. Okay, and see whether it will work or not. So you can just press up arrow key and you will see the history of the earlier commands that you type. You don't have to type in again the same things. Okay.
Arrow.
Yeah. So you can see that now this command has been run properly, and 2 new results are there in the workspace. You can see the result and list result. So what is there in the result? The result is it is the gene that we inquire for, does B0116. Okay. So in the E. coli core model, no reaction is there associated with this gene. The gene is there, but they haven't used it in any reaction.
Abuse. Can you show the previous command? Just
This is the command, right?
the just previous one.
Which one?
This one.
This was the
Model override. Okay.
this I printed because I wanted to show that the original model file is also there to differentiate between the original model and the core model.
Underscore ORI is goes to enter.
Core model.
Yeah. Okay.
Okay, and we can go here and check. In the result, this is the gene that we inquired for, and in the list result, nothing is there. It means it is not associated with any gene. Okay, so we will try to inquire for a different gene. Say, first I have to find out which genes are there in this new model. So I will try to print first 10 genes for the model. Model.genes 1, 2, 10.
01 का 01 में डालना.
So these are the first 10 genes that are present in the E.coli core model. So you can randomly take any gene and find out with which what reactions it is associated. Okay, so I will take this 474 gene. You can take different than mine and see. Is it? Yeah, it is not also associated with any reaction. 485. Oh.
I'm able to get to B011 reactions. I'm able to get those.
For what 116?
Yeah.
How?
Column 1 through 4, PDH and the simplest aspect like glycolysis. In that model, first model, original.
Yeah, you are right. Actually, it should be there.
I'm able to find reaction.
I will try to rerun the same thing, right?
Right.
T011.
116 तो आ रहा है ये देखो.
Model grade like again model.
Very good. Very good. Very good. Outside it.
यार ये था
Yes.
ये देखो इसमें क्या लिखा था मोड 1 है ना ये मॉडल है मैं मोड 1 कर रहा था क्या है
उड़ रहे हो तुमने किया होगा ना
जो किया हुआ अब ये समझ में आ रहा है
तो ये कैसे इनको बनाया किसने बनाया ये इनको
हमें कोई एकदम ये जो है ना ये सिर्फ ऑटोमेटिक आता
ये भी रिसर्च होगा ना
है पेपर पब्लिश होगा ना लिटरेचर में उसमें से उठाते हो
Okay, so the problem here is that I was not entering the other optional arguments
0 and 1
0 and 1.
बाइनरी लेकर
Yeah, I have changed those things. Random.
And
So if we
nor, and
enter the optional arguments,
column 4 through 6.
So what are the 0 and 1 which you
Binary code.
no, right now I'm not clear what is this argument about. I will let you know again. Okay. So now we can see that these two reactions, these two reactions are associated with this gene. Okay, B0116. And what are those two reactions? Pyruvate dehydrogenase and 2-oxalobutyrate reactions. Okay. And it is also showing the reaction structure and
Related to fermentation.
in which reactions they are included. For example, the first is included in the citric acid cycle and the second in glycolysis.
But Piyush, we cannot do this to the previous model, right?
0-1 डाला आपने?
0 नाम दिया।
यही है मेन फाइंडिंग। देखो करके
Right. You are asking something?
We cannot do this to the previous model, right? Like to extract the reactions from the gene. You require that field GR rules for extracting these things.
This is not
Or
coming.
like that field is necessary to use this command.
Yeah. Because we have to give this thing because genes need to be associated with the reactions. Okay, we have to give in model itself doesn't know anything. Whatever we will give it, it will print. So yeah.
Yeah, I'm not getting this table.
I'm not getting this.
Did you put the optional arguments 0 and 1?
Yes,
You tried it?
it's showing invalid expression.
फिर इनकी कमांड देखते ना
Write this. No, put in a comma before. Put in a comma before
once
0.
they show that slide like your command
Put in a comma before 0. Everyone else got it? Thanks.
So comma for 0 and 1 separately.
Yeah. So
Okay. I got.
B हाँ comma 0, 1 बस
So next thing is we will try to print the reactions IDs. Okay.
ये इसको यहाँ पे खोला भी आता है
Yeah.
Reaction IDs.
ग्लाइकोलाइसिस है और फिर TCA
And the command is find
network through this
reaction ID.
ये सब MATLAB करता है
अपने पास है ये सॉफ्टवेयर
डिपार्टमेंट
Model. Model dot reactions. We are trying to print first 15 reactions. Okay.
model.
And so it is just printing the serial numbers of the first 15 reactions.
instead of 15 we can take
And
any number
समझने के
model reactions
if we want to know the names or the identifier of those reactions, we have to just use model dot reactions.
Okay.
1 to 50. So these are the
okay.
first names of the first 15 reactions
देखो model dot reaction
or the identifiers of first 15 reactions. Are you able to follow?
Yes, one thing. Sometimes we put 2 columns, 2 brackets, not column.
Okay.
Like
So-
I put 1 bracket and it's showing the error, and then it's suggested to 2 brackets. So when we use 2 brackets,
For example, here I have used 1 bracket. We have to just complete the whole thing, right? Here we have started the bracket, then we have to close it. Whenever we start a bracket, we have to just close it, right? So, for example, in this case,
1 to 15 वाले में हाँ.
Yeah, I was trying to find the reaction IDs of the first 15 reactions. So this thing stands for first 15 reactions, and I want to know the IDs of these first 15 reactions. Okay. Therefore, I am giving this as an argument for the command find reaction IDs. So therefore, I have to close this bracket also.
Okay, okay.
That's the thing. Okay. So I can also like that we did for the genes, we can find out any idea of any specific reaction also using the same command. Find reaction IDs and put in model. And for example, the enolase reaction we want to know for. So it is printed as 18th. So it is
18th reaction.
It is the 18th reaction in the E. coli core model. Okay. So similarly, the same thing can be done.
For simple arithmetic calculations as shown.
So this we can see for any reactions, right? Na? Suppose इसमें तो इसने बताया enolase is for 18th one.
Yeah.
Any other reactions we can also see.
Of course, you can see any other reaction. Just you have to type the correct identifier of the reaction. Okay. Similarly, we can find out the first 15 metabolites. Model.mats. 1 to 15. And these are the first 15 metabolites that are there in the model. If you want to find out any specific ID of the metabolite, ID of any specific metabolite, then use the command
Because to find
find
ID.
met ID
Model one.
we have to put the correct name of the model that we are using. So we are using the E. coli core model. So I'm typing in model. So we can choose any metabolite here in the list above. Suppose we are searching for ADP. ADP cytosol. Complete the bracket. So as you can see that it is the 13th metabolite. 13th, 14, 15. As we have printed the 15 metabolites in the serial order.
13.
Okay.
KG.
And if we want to print the name of the metabolite, then we have to use this command.
Model.mat name.
Model.mat names. And just put in the ID. So for example, what is the ID here? 13. And the name of the metabolite is ADP. Okay. And the next command can be used to just print the formula. So this is the formula of ADP. Okay. So these were some basic attributes of the model. We can also find out the upper bounds and lower bounds using any. For example, I want to find out the upper bound of and I know the ideal. For example, this 13th metabolite, I want to know what is the upper bound. So the upper bound is 1000 for the 13th reaction. And if I want to print the formula for a reaction, use this command. In reaction formula Model Inoles. Not available. So this Inoles reaction is not available in the E. coli core model. But here it is printing.
So I have a doubt, like in the initial command you entered formulas.
ENO is the capital, right? That's why it was showing error. Yeah. ENO. Yeah, what was the question? Sorry.
So in the initial command you entered the model.met formulas, and over here you have mentioned formula. So like, why is the difference?
Yeah, 2PG H2O plus PEP.
MAT formulas. Okay, and here.
Formula.
So basically it is it's predefined. It is not my choice here. It is defined like that in the COBRA structure. We are bound to type in the way it is defined. Okay. Yeah, any question? Now you have done some basic exercises. You have understood how the model is designed or defined. Yes, ma'am. Yeah.
She can ask you.
Can you please just ask your question?
Okay, thank you. Now, I just wanted to check, is there some possibility where we, or list where we can find these commands? I mean, because maybe we will not be able, when we were practicing, maybe we will be wrong or forgot something. Is there somewhere where we can find kind of help, or how to know all these commands, when to use, which one will help us how to extract them?
Okay, ma'am. Do we have your email IDs? Do these organizers of the workshop have got your email IDs?
Yeah, I suppose they have.
Yeah. So what I will do, I will just tell them to email this file of this booklet, the PDF file of this booklet or the Word file of this booklet to your email ID. Okay.
Okay, great. Thank you very much.
So in that way, you will be able to practice these commands.
Yeah, thank you very much.
Yeah. And I will also tell some additional online resources from where you can learn more about these commands. So you will be able to do much more things than whatever we are doing here. Okay. So I'll just try to clear my command window. So another important command is check objective that I want to tell you. Okay, any guesses what this command might be doing? What will be the function of this command? Anyone can guess? Answer is, can you see the answer on your screen? What is the answer? The command is there on the 26th page number. Check objective, then in model, then in brackets, you have to put the name of the model. All right. Now run it, press enter. So this command check objective, as you have learned in your lectures, the Professor Raghunathan explained very well that in FBA, FBA is a linear programming based problem.
Yeah.
So in any linear programming based problem, the problem is solved in a way that we have an objective to solve that problem for. Okay. Since we are doing here FB only, we have to define our objective. Okay. So in this case, in the case of flux balance analysis of the organisms, we set the objective as the growth of that organism. Okay, the default objective. Yeah, we can change it, but the default objective is set as the growth, and growth is estimated by using the biomass reaction. Okay, so biomass reaction is 1 second. Biomass reaction is the reaction. You can see that here. In the summary of this reaction, you can see that there are these metabolites and their coefficients. Okay, so this reaction is having these metabolites. Display all 23 rows. So unable to display because it is no longer exists. So I will try to get it from here. Not sure. So, so it is not able to show the whole 23 rows. So basically, you can understand from this data also. The main logic is that while constructing the biomass equation, you measure all the biomass composition of the organism. You just grow the organism and then analyze the biochemical composition. And you, when you identify what is the composition, you give the relative coefficient of these metabolites, and in that way you make the biomass equation. We measure all the biomass composition, like what are all the amino acids, or what are the different fatty acids, and what is the carbohydrates that are present in the biomass of the organism. And in that way, the biomass equation is formed. Okay. So for different kind of organisms, the biomass equation would be different because they will be having different biomass composition. Okay. But for similar organisms, for example, E. coli, we have different strains of E. coli. The biomass composition will be relatively similar. It won't be varying much. So we can use the same biomass equation for similar or identical, for different strains of E. coli also. Okay. So usually the modelers use the same equation for while making a model for different strains of E. coli.
Piyush, we are not getting this again showing some simulink.
You have entered something wrong. Check objective. CHECK.
I put O capital.
Yeah.
I put capital also.
Try to put capital O.
I put here.
Model requires. You have deleted the model, right? You have
Workspace cleared.
Yeah, you have cleared your workspace. Yeah, load the model again into your workspace. Okay, so she asked that can we change the objective? Yes, we can change, and the command for that is change objective. Model. We have to define.
That's called model.
And now I will put the name of the the identifier of the reaction that I want to change the model for. For example, if I want to change the model example, for example, ethanol exchange, I want to maximize the production of ethanol. I want to set the objective of the cell that you have to produce more and more ethanol. So this thing will do, it will divert whole of the glucose flux and it will divert the maximum flux towards the ethanol production. Okay, so I will just have to put in the name of the ethanol reaction, exchange reaction of ethanol. And how can I know what is the reaction identifier of that ethanol production? I have to check here. I'll go into here and in the reactions, and it should be there in the exchange reactions. But I don't think that, yeah. Suppose we can take this also, exchange of succinate. Okay, this is succinate exchange reaction. So we can change and we can take any reaction and put it in the place of the biomass reaction and see what are the results. This okay. So this is acetate exchange, says CO2, and this is the ethanol exchange reaction. Okay. EXETOH. EX underscore ETOH. Yeah, we can copy this also. Okay. So objective something. Okay, I have put C there. Was it C or E there? E. It was E, right?
Yeah. So you just copy.
So it's the X capital over.
Right. Thank you. Okay. So why it is printing the model attributes again? Because it has updated the model. We have updated the model. We are defining our new model with the objective reaction as ethanol exchange. So our model has been changed now because earlier the objective function was biomass reaction. Okay. Are you able to follow? Try to tap in with your own hands.
Model equal to
Objective model.
Are you able to do that?
Model.
Piyush, can you have two objective functions like you want to maximize the production of two metabolites in the metabolism?
No. You cannot have two objective functions.
I want to maximize production.
You have to again, I don't know how to do that because in this way you cannot do this.
Okay, this we cannot do. But
Maybe you have to design a function for this.
Yes, now we got back. So can you repeat it?
Which thing?
The last one
How
because
to change the objective?
Yeah, you have to just type in this change.
so how we okay, what is the basic fundamental for changing the objective? Like we have to plan some reaction and then we have to put that reaction name for change, or why?
Yeah, I have used this reaction, ethanol exchange. I have taken this reaction from the list of the reactions that are already present in the model, that are already present. So I am just changing the objective function from the biomass reaction to the ethanol exchange reaction.
Okay, so suppose we have some objective, like we have to study this particular pathway, the particular reaction. So we
Right.
put that one here.
Yeah, if you want to maximize the production of that reaction, you want to maximize the flux of that reaction, you have to put that reaction here.
Okay.
Yes, ma'am.
What is the meaning of result which has come now? What do we understand from this?
From this thing,
Change of.
ma'am, this is nothing. It is printing the model again. It's just printing the same model attribute.
Showing that if the objective is ethanol production, then these are the reactions. This is about if it's showing
the
reaction
model is being
Modified.
Same जैसे ethanol का था वैसे ethanol का.
It is the same model, but the objective has been changed. It is not shown here. If you go there into the model, and there you will check, you will find that Now again check, print the command check objective. Type check objective now, and it should show the ethanol exchange reaction. Type in the command check objective. So now it is showing the ethanol exchange as the objective. Okay. Right?
the table is coming and then it is written
Yeah.
and reactions number 24.
Yeah. Reaction number 20.
This became our objective.
Objective of the model is now ethanol exchange.
Accordingly, the other files have been modified accordingly.
No. Nothing has been modified. We have just updated the objective of the model. Initially, what was the objective of the model? To maximize the growth. As the Professor Raghunathan, you have attended the lecture. What she told that this is the theory that the organism tries to grow maximally, and to grow maximally, it has to produce its biomass. So it is accordingly we put the objective function of the model here, the biomass reaction. So when we provide glucose, it will try to maximize the flux towards the biomass components. It will try to maximally produce the biomass components. Okay. So more biomass, the more will be the growth rate. Okay. This is a general case. As we assume that the organism will try to maximize its own growth. Okay. But in our case, we humans, we want to produce more ethanol. Our objective is different. We don't want that E. coli to grow more. We want that E. coli to produce more ethanol. So we will see that how much maximum flux can be theoretically diverted towards the ethanol production. So we will see that with all that metabolic network the E. coli is having, and we provide the glucose intake rate, what can be the maximum yield or the maximum flux of ethanol he can theoretically get. So for that, we have to change the objective function. Okay, when objective function is changed, and we will do the flux balance analysis or the simulation. So we will be solving a problem where the objective function is ethanol production, and it will try to maximize that particular reaction. Okay.
That part will now come.
Yeah, that part will come now.
Just terms of mathematics, I can consider ethanol as metabolite. So you're looking that objective to the ethanol. So when you come to the big molecule, like protein, so will you define all amino acids or just any biomass?
So there is no single reaction for protein production.
Right.
Okay. We are not having any reaction for protein production, for a particular protein production here. We have to add that kind of reaction.
So is it related to the amino acids?
No, if we know that these are the amino acids that are present in this protein, we will try to maximize those amino acids, for example. But we cannot do that in one shot. We will try to maximize all the constituents, but we cannot do that in one shot.
Right.
If we can maximize one reaction at a time, we cannot give multiple objective functions. That problem will be a different kind of problem. Okay. Right now we are solving a linear programming problem that can become a mixed integer or a more complex mathematical problem. So we are simultaneously trying to maximize more than one reaction. It will be a complex problem. Okay. We are now doing the simple problem, simple case. This is the case.
So it's more complex, but in that ultimately comes with the amino acid reactions.
Absolutely.
Right. End product is the amino acid.
Yeah.
And combine and give the yield of protein.
So now we will. So as we have changed our objective, we will again change our objective to the biomass reaction and see what is the growth rate that is it predicts. Where is the name of that biomass reaction? So I have to get the name of the biomass reaction. So here I will copy the name and again put it here in single quotes, and I will change it again. So now I have re-updated the objective as the biomass reaction. Okay, so the model is again updated to its initial value. Now to do the flux balance analysis, we will put the command FBA solution and the command is optimize the model. The model we are using and what is our objective? We want to maximize the objective function. We want to solve the problem like that. We want to maximize the objective function. And what is the objective function here? The biomass reaction. Okay. So FBA solution here is, it is a variable. We want to store the result of this this command into the variable FBA solution. So this is the solution. This thing F, as you can see that it is showing F equals to 0.87. Okay, so this F is the flux value of the objective function. Okay, the COBRA has solved the problem and it has found out that find out that the maximum flux of the objective function can be 0.87. Okay, this is the maximum flux. It has tried to maximize the biomass reaction. It has found out that this is the maximum growth rate that can be achieved with how much of maximum in glucose intake rate? -10. Because we have, I have shown you earlier that what was the lower bound that we have taken was -10. Okay, so this 0.87 is the growth rate. And what are the other fluxes? It must have also calculated what are the other fluxes of the reaction in the model. So how to look for that? We will just go into this solution. Okay, and this is our objective function, and this vector V. Yes, this FBA solution V, it is showing here the values of the fluxes of other reaction that the COBRA has calculated. Yes.
I'm getting the F value as
You are getting F value as 20.
Same here.
Same here. Yeah.
I'm not getting it.
Okay, let me see.
ये नहीं है वो maximum
Anyone else is getting F value as 20? Anyone else? How many of you are getting F value 0.87? Are getting 0.87? Can you see why is she's getting 20?
I've done the same.
He has entered the same thing.
The objective.
Previous wala
You have changed the objective again. Have you?
Yeah, I've changed it to biomass.
You have changed it again.
अब तो आ ही नहीं रहा है ये देखो
ये देखो
Are you doing it? It was not changed properly. Just try to update it again.
वो full वो equals to equal space दिए दो
No. You have to include the model also, right? Because we haven't model. You have to
solution है क्या solution
solution
model is just to change.
solution
optimize CB model का M capital
minus
CB capital
C capital भी छोटा है ना
M बड़ा
to DB model
हाँ वो close bracket
Yes, sir. Are you getting now?
अभी enter मत करना बता दो comma semicolon max और रुको अच्छा करो यहाँ space
Right. Yes.
enter
यहाँ space लगाओ
Solutions, solve Cobra,
Let me see.
उसको दिखा देते हैं और same command
MPS reduction optimize
Yeah. Press enter.
Cobra Toolbox initialize करिए ना.
Did you initialize the Cobra model?
so silly
You put initialize the Cobra model?
So उसके बाद कैसे?
plus immediate
Do it again. Init Cobra Toolbox. Init Cobra Toolbox. This command. Box.
नहीं तुम कर रहे हो तो मैं वहाँ
Box. Then in bracket, put in false.
लैपटॉप में
बिल्कुल बिल्कुल अब करना यार
initialize this.
क्या वैल्यू आई 0.87 ना
हाँ
I did that
okay we
Very early.
0.87
for running all these models.
आपको 20 आ रहा था.
नहीं
First thing you do is initialize. Thanks.
मैं आपको ये नहीं आता कैसे लिखा है
सर आप अगर आर्ट पे करने से नहीं ज्यादा इजी पड़ता है क्योंकि इसमें सजेशन दे देता है उसमें सजेशन नहीं देता है.
आपने कहाँ से लिया है?
आर्ट में मैंने क्यों
नहीं ग्राफ वगैरह बना
Something.
सर
वो वाले होते हैं ना क्या कहते हैं पीसीए एनालिसिस
Model मतलब
इसी एनालिसिस ओरिजिन से वेटर से कोई नहीं करता है. ओरिजिन में मैं करता हूं.
ओरिजिन मेरे को बताना है
ओरिजिन जो तो स्पेसिफिक एप्स आती हैं उसकी आप अलग-अलग जैसे इसका डेटा है उसका पीसी कर देगा. ऐसे इन जनरल भी कर सकते हैं. उसके बाद प्रीडिफाइंड मॉडल्स होते हैं.
So now you are all able to
कि जैसे मेरे पास मैट्रिक्स
exchange for exchange for that thing that is
basically what the formula you have not initialized the Cobra.
उसमें सामने दिया है.
Yeah. Have you?
The objective first.
But
This one, this one.
But then you have again started the MATLAB, right?
That by default should the biomass.
Every time you have to
Should the biomass.
start the
Table
MATLAB.
should. But if
You
you
have to put
but if you
include the Cobra.
Then
I also did that.
start the mixed exchange. Then we again change it by in it. तरीके से
Why did
In the false.
ethanol is like a very desirable product in Gap. Basically, I want to sign up and trade.
But we started the
जो
matter.
उसका जो ग्राफ है
So they have
Produce ethanol. आपको बड़ा कमर्शियल देना पड़ेगा।
right.
तो point ये है कि
क्या नाम है इसका ओरिजिन?
किस फाइल लिखते हैं
right now we are not focusing on ethanol production. That's why we changed the objective. Is trying to grow the cells.
Because we are using the variable name model.
We have seen some kind of window के लिए.
Then it will calculate directly.
Absolutely right. I mean, that's
Yeah, now it's printing. Are you getting it?
Yeah. Most of the genetic engineering.
Now it's getting.
But we just try to understand the metabolism.
So everyone is getting the growth rate 0.87 now.
Yes.
Understanding.
Okay.
Right. Like commercial.
Are you doing also? We're entering the commands, right?
So is this specific group
Title, we also try to
Yeah, this is the specific growth rate. Okay,
which does not
use the biomass.
which does not change
is a constant and does not
with time. Yeah, in the log phase. In the log.
specific group rate.
Yeah, in the log phase. So
What is the F1 and F2?
Yeah, F1 also and F0, F1 and F are the same thing.
So these are also different attributes, but our main thing is we have to look for objective function and these fluxes B.
Can you explain this V, Y, W and S? What are these?
I was about to ask this. Even
If you want to know what all these things are, okay, so you can read here. Just evaluate or open the selection or help on the selection. Okay,
इसमें बताएगा.
so here you will find what are the inputs and what are the
Research.
answers that it can print. Okay.
F देखो linear.
Yeah, F is the linear objective value, F naught is the 0 norm objective value, 1 norm objective value, 2 norm objective value. They are some parameters of importance, but not of much importance right now.
अपने कहाँ दिया objective box?
तो change किया तो biomass से growth rate.
Yeah, the important thing is this thing. Stat value. If the stat value is 1, it means that the solver has solved the problem correctly and find out the optimal solution. Okay. If the stat value is 0, the problem is infeasible. It is not able to solve the problem. Okay. So what in our case, stat is 1.
Yes.
Can you see that?
Yes.
Stat is 1. So it means that the solver has found out the optimal solution.
Okay.
These are the things important things that we have to use. Okay. Other things are not of much importance. So now you again change the objective function to ethanol exchange and rerun the FBA solution command and see what is the optimal solution you get. You should get 20 that she was getting. Okay. Try to do that with your own hands. Change the objective function again to the ethanol exchange and
Model.
Objective.
Model. Exchange ये तो अपने ETO
Exchange कर रहे हैं ना.
अब क्या करना?
you can simply do that by using up arrow.
Yeah.
Okay.
And now the value is 20.
20. Yeah. For example, I now change the objective function again. The model has been updated with the new objective. I can check the objective.
अब मैंने से कर दिया.
So now the objective has changed to ethanol exchange. And now I will again calculate the optimal solution using the optimize CB model command and try to maximize the production of my objective function. And you can see the maximum, the optimal solution for this objective function is 20. Okay.
What does it actually signify? I'm not getting that.
Sorry.
What does it actually signify? Like 0.37 from the value in the previous one. That is 20. So what should we interpret?
Yeah. So this is a very good question. Everyone should listen this question. Yeah. So he is asking what is the meaning of this F20? The F is our F is the optimal solution for the objective function, and 20 is the answer. And the units are millimoles per gram disulfide per hour. So it is basically the flux value of the objective function. Okay. And what is the objective function? Ethanol exchange. Okay. So the model has calculated that if we provide glucose with integrate of -10, with the integrate of 10, and we maximize the production of ethanol with no growth at all. Okay. We try to maximize the production of ethanol. So what is the maximum ethanol that can be produced? All of the flux, if we are diverting towards the ethanol exchange reaction, what is the maximum ethanol that can be produced? So the answer is 20. You know? So you can also think it with the help of the metabolic network that Shirish sir also explained, that what is the theoretical yield from 1 mole of glucose, how many moles of ethanol can be produced? 2. Okay, theoretically. So if we provide 10 moles, how many moles of ethanol can be produced theoretically? 20. So this is the answer. This is to answer your question. This is to answer your question. Okay. So what are the other fluxes with this objective solution? If we want to know what are the other fluxes, I will go here and see. And you can see here that the 13th reaction was the biomass reaction, and it is having an answer 0. What is the meaning of 0? That no biomass is being produced because we have diverted all the flux towards the ethanol exchange reaction, right? So how the cell will produce the biomass? We can do this thing on a computer, but is it possible practically to divert all the flux to the ethanol? It is not possible. The cell has to grow, at least with some growth. Okay, so you can also understand that the maximum growth rate that was predicted was 0.87. In a real biological scenario, you will not get this growth rate. Okay. If the biomass reaction, F is the objective function. If the objective function is biomass reaction, F will be growth rate. If the objective function is ethanol exchange reaction, F would be the flux of that corresponding reaction. Okay, F denotes the flux of objective function that we are choosing. Sorry? No, the flux of the objective function.
इथेनॉल के केस में फ्लक्स तो खैर सबका यही है.
In which Excel table?
When you're going to FTA solution and we go to B.
Video में
Which table? Tell me.
अब तो वैसे आ गया था.
This table? No, these are the fluxes of the reactions that are there in the model. All the different reactions. You can see, right? Huh? Yes. So, this is the flux of these reactions. If I copy this column, okay, and paste it into a Excel sheet. And now I will copy the answer. So these are the fluxes of these reactions, okay? The flux of the biomass reaction is 0. And all the internal reactions are also shown. And what is the flux here of the glucose intake? It has taken the maximum allowed value that we have put. What the maximum allowed value we put for the glucose? -10. And you can take this maximum value. And so the solver has taken the maximum allowed value. Okay. So can you see the oxygen? Can you look at the oxygen flux here?
Yes.
I think this is the.
Yes.
So this much oxygen it is using. Yeah. It is not using the maximum allowed value. It is using only this much oxygen. 1.33 into -1.33 into 10 to the power -31. So very little oxygen it is using. And the minus sign indicates that it is taking the oxygen from the environment. Okay. So able to understand? How many of you are able to understand this thing? Is it clear?
Yes.
Yeah.
Oh, thanks. Now, since we allowed, what was the maximum allowed value for the oxygen uptake? It was -1000. Okay. So we want that we don't want any oxygen. We are not. We want to simulate it for anaerobic conditions. So what should be the condition for anaerobic condition? What is the condition for anaerobic?
No oxygen.
No oxygen means the oxygen intake value is 0. Okay. So if we want to simulate the model in that condition, we have to fix the lower bound of the oxygen intake reaction as 0. Okay. So we will do that now. We will update the model and use the command change reaction bounds. You can also see that command on the page number
29.
Yeah, page number 28. Reaction bounds. Then you have to use the name of the model that we are using. Then we have to use the name of the reaction that we want to change the bound for. O2. As I have entered the name that it is capital X, right?
O2E.
E. And I want to fix the lower bound. Okay. 0. And I will use L as the for lower bound. Okay. Some error is
Extra bracket. You didn't close the, yeah.
okay. I haven't closed it. Thanks for correcting. So the model has been updated with the lower bound of oxygen as 0. So oxygen cannot be taken now. And I will try to optimize now. Yeah. So the value is again coming 20y because I have not changed the objective function. It has taken the objective function as ethanol. So there's now I have to re-update the objective function. Yeah. I've used this command model change objective again to the biomass reaction. And now I will do again the. So now the growth is coming as 0.21. So as we know already that in anaerobic condition, the growth is less than the aerobic condition. In aerobic condition, large amount of biomass is produced than the anaerobic.
Stat value 1 is.
Stat value is also 1. It's showing 0.
What is the objective change करके
Again हमने उसका objective पहले हमने change the objective as ethanol exchange. Now we have again changed the objective as biomass reaction. Okay. If we want to see how much a cell is growing, the growth of the cell, we have to set the objective as the biomass reaction. Okay. So this is the anaerobic growth rate. So these things we were doing for the E. coli core model. Can we do this thing for the IGO 1366 model? How we will do that? We will just change
the model name.
And use the name of the model of IGO 1366, which we have saved as model.ori. Okay. So here the growth rate is 0.98 maximum growth rate. Okay. It is a bit different. Why? Why it is a bit different? Because it will be having somewhat different biomass reaction. They might be using certain different biomass reaction. That's why the growth rate is different. Okay. Now check what is the if we change the objective here, what is the maximum ethanol that can be produced? Or
To what?
check what is the anaerobic growth rate for the IGO 1366. We update the original model. No, we won't do it for the original model. We will keep our original model intact. What we will do that we will make a new variable, model 1, and we will equate it to the model original. Okay. So I have created a new model variable model 1, which is equal to the original model of IGO 1366. And now I will change its bounds.
Model 1 में यहाँ पे भी वो है.
Bounds of this as lower bounds 0. Okay. And now I will calculate what is the maximum growth rate with which the IGO 1366 model can predict. Okay, it hasn't changed. Why?
It is equivalent to original model.
Okay, because there is no reaction of this name into the model. So we have to see with what name the oxygen uptake reaction is defined in this model. Okay. So we have to do it manually. And some people have started to exhaust. I'm seeing.
I see.
Okay, so we have write this file. Didn't we? These are the reaction lists of the IGO 1366 model. We can search here, and it is quite easy for us to search here for the oxygen uptake. We'll just find Where is oxygen in this column?
It 1982.
1982.
Row number.
Yeah, it is the row number. Okay.
Row 2 TEX.
Oxygen. Input control input oxygen will show. It's not showing. That's the. Now you have to take that. It's not showing. It's showing me it's not. Oxy put oxygen. OX put. OX.
But it is oxygen transport via diffusion, right?
Will show the all the X in everything.
It is not an exchange reaction.
EX copy.
The notification is coming. TV will soon turn off.
Which model?
Press any button on the
Right. Yeah, 156. 186.
Transport via diffusion periplasm to cytosol.
This is the main model. Reaction list of 96.
Right. This is the reaction from periplasm to cytosol. OTP.
Exchange reaction only is searching, right?
Yeah, and
Okay. So here you can see that in this reaction, the oxygen is being transported from periplasm to cytosol. Since we have to transfer the oxygen into the cytosol, only then it will be utilized. So we have to use this reaction. Okay, and then change it. So I will copy this reaction, Control C, and again I will change reaction bond. And now try to do the. Oh, it's still not working. Okay, leave it right now. We will see it after the break. What is the time? Okay, just have a 5 minutes break. Sorry, 10 to 15 minutes break, and then come back again. Okay, we will again start at around 4:10. 15 minutes break for tea. Yeah. Yeah, we are having a 10 minutes break for the online participants. 10 to 15 minutes break, and we will again come back here. We are going to start from here. 6 people are there. There is a 2 comments. Reaction ID 185 is the oxygen exchange. Reaction ID 185. Okay, let me see. Thank you for the person. 185.
$186.
Yeah, exchange O2, say 185.
Yeah, actually it is 186 to be precise. Thank you the person. Now we can do it.
First row is header.
First row is
Header.
header.
85.
No, it is
186 ही है क्योंकि फर्स्ट रो जो है वो हेडर रो है, तो इस वजह से 186 हो गया है.
okay. You're right. So reaction ID is 185 only. Yeah, he's right. I was wrong because this row is header. So the reaction ID for the oxygen exchange is
185.
185. So we have to copy the name of the reaction. Sorry, the name of the identifier of the reaction. Control C. All of you can do the same thing and try to simulate. Change the reaction bond of this reaction. To 0. And then try to maximize. Have we changed the correct one?
0.98. One.
वही आ रहा था I need start book.
It's showing the one.
Yes, सही तो ये रहा.
बच्चों के इनका देखते रहेंगे.
3.98.
Influence करने का तो
Okay, come back after tea break. I'll see what is the problem.
Okay.
Before that show the same thing. Before giving the constraint, goes the same.
Yeah, please go for the tea break, and we will be together again at 4:15. Please, we'll have much to do. वो प्रॉब्लम पता चल गई मुझे. ये हुआ. अब आ जाए.
बॉन्ड वाला ना ये जैसे हुआ ना ये वाला
Yeah. M.
So is it fine?
Yeah. You have your food.
Light is very low.
Like
I'm blind.
I keep low because
Yeah, the other strain, but what about this strain?
but I can use.
Yes, sir.
Yeah, उसको पानी अपना बार बार दो.
तो इसमें गलत हो गया है।
So now what is this?
वो मॉडल 1 क्या था वो
जो इक्वलाई का जो मॉडल हम यूज कर रहे हैं ना IG-136 उसको मैंने डिफाइन कर रखा है मॉडल 1 के नाम से
और यहाँ पे तुम
और यहाँ पे अपडेट कर रहा हूँ दूसरा मॉडल।
अच्छा अच्छा अच्छा
तो वो उस मॉडल को अपडेट कर देता है।
अभी इसमें क्या है, इसमें आए हैं मॉडल 1 से, so still I'm getting the 0.98. Yeah, base solution 0.98 I'm getting. ये वाला तो optimize 1 होते right?
The problem is that here you know use the updating model.
Oh, right.
I was doing the same thing again and again. So that was
So now I have to do model 1
and then
if we
yeah now you will get that.
right. Actually end to end पता नहीं चलेगा एग्जाम तक।
तुम बैठ के कर लो ना
हाँ हाँ अभी बैठना ही पड़ेगा।
वॉल्यूम चल रहा है
In between time चाहिए तो you take 5 minutes. Don't continuous. You don't need to.
मैं कभी at least we need 1.5 hours outcome के लिए।
बैठ के थोड़ा आराम आराम से कर दो कोई ऐसा थोड़ा
Internet. Right.
Zoom close कर दिया।
नहीं
Don't
mute it.
core model or
Model underscore 1 equal to change rate in long model.
we converted that
X to be 0.
model 1 we took the variable model underscore 1 model 1. So the model is
Okay, so now
X.
D D है ना?
4 model 4 okay 4 model is done with okay
we have simulated this model in 2 conditions.
उसके बाद में क्या करना है? Study solution optimize model 1.
फिर ऊपर वाला ऊपर को दूसरा पहले वाला को
okay
क्या क्या क्या
situation okay change objective
3.98 to 4 hours.
check objective it is showing the core
अब नेक्स्ट क्या बोला तुमने?
core okay
So we were getting this error. We were not getting the correct growth rate because we were not updating the model in which we have saved our IG-136 model. The variable we were taking here as model, but we have saved our IG-136 model as model underscore 1. When we update the correct model file with lower bound 0 of the oxygen exchange reaction, and then again we do the optimize, we run the optimize CV function, the growth rate is calculated as 0.24. Okay. Okay. Yeah.
it's showing the same value for me 20 not 20 it's showing 0.98 0.9
It's showing the same value. Are you using
FBA मॉडल 1 भी करना है उसको?
FBA model 1 भी करना ना वहाँ पर? You have to use here model 1 also. Are you using the same thing?
I'm using the first model which we did before the core model like the IJ what is that IGO
बंद करना
IGO yeah that model
So you have to use the same thing here.
Yeah,
The model you have updated and the model you are running FBA for.
yeah, I mean, that's what I'm doing.
Try to do it. It should do that.
No, you have to copy that oxygen. I think you
Yeah,
must be-
oxygen, I have given a constraint giving zeros lower limit.
As soon as getting the same thing, you are able to get the growth rates 0.24. 20. Just update your objective function. Yeah.
So like I had constraint as, you know.
Yeah. Model. What is the model file here? Can you just change the layout?
नवीन जब अपन नई चीज को डिसाइड करेंगे तो मेरा क्वेश्चन ये है नए काम को हमने स्टार्ट किया तो
Already they.
So what is the model here?
That's the IJO.
IJO 1366 model.
Yeah.
And
since उसके
IMA selection is this you have copied from here only?
But why don't we,
Yeah,
So,
so I'm giving a
It should
specific.
it is not a problem. You can use B or L. B means you are just fixing both upper and lower. I will tell.
That's the important model.
but again, it is using model 1 or model.
Okay, I think
बता दे रहा है
ये सारा बता
zero or O, I think. Oh,
without
made a mistake.
the काम
Just go
Yeah,
for the
yeah.
model
There
underscore.
is
Decompose.
mistake which I made. I think that's zero.
और फिर इसका ये
Zero.
इसका use
Have O here.
I think that's so. That's why. But I have,
No, but he is.
Actually, he's using same, right?
I have done 0, 1.
Table with the idea.
ये
Have you-
kind of
I have done 0, 1.
एक्सपेरिमेंट का ही पार्ट हो गया ना
Name
MATLAB
model.
Yeah, it's coming now. So I got then
FBA.
right. Max.
Okay.
Okay.
Okay. So the first
Go to
one,
the
right?
model
Go
one.
for
This
that model.
you
And the
are
fixing.
submitting for model underscore 1.
Show model.
Yeah, you have to change
Go to
the
the model
model.
underscore one.
You have to give underscore 1.
Where FBA.
Model change bond ratio. Here,
Yeah.
you go for the model underscore 1 is equal to change bond.
Okay. Model underscore 1 equals
Yeah.
to change reaction 1.
Yeah,
Model
right.
underscore 1.
Right.
Okay. Okay.
There you just then FBA. Enter FBA. So,
So everyone has emailed the soft copy of the booklet
head with
to their email. You have your soft copy on your desktop, right?
This is 0 and this is 1.
This is
Okay.
L not 1.
So I am typing model underscore 1.
LRNE like
Okay.
value
And pressing enter. So are you also
you go
check the email, check the model ID. What is the model ID on your screen? Or the model underscore 1 variable? Is it IGO 1366 or E. coli core?
give any of the letter.
IGO 1366.
So what is
Okay. Everyone is having the same thing?
Okay.
Right, certain commands.
Just type in the name. Type in the name of the variable. I want to make sure that we all of us are using the same model. Okay. Core model is coming. So do one thing. Make type in model underscore 1. Type in model underscore 1 is equal to model underscore ORI. Do you have model underscore ORI on your workspace? And make it equals. Those who are having model E. coli core model coming as model underscore 1, re-update your variable to model underscore ORI. Have you done?
I have a question, like in the last command that we did for the model one, we used the upper bound as with the character L. Now we have written B. So why is it?
Yeah. So in the booklet, it is written as B, right? So B means if we want to fix both upper and lower bound, then we will use the character B. Okay. If we want to just change the lower bound, we will use the character L. And if we want to change the upper bound, we use the character U. And if we want to set both upper and lower bound, we want to fix it. We want to give it a fixed value. Neither go up or down. Just take this value only. Okay. Then it should be used B. It should use B. Okay.
Sir,
You understood?
no.
No. Okay.
There are like lower and upper bounds both if we are.
If lower and upper bounds are two different values, minus say lower bound is 0 and upper bound is 1000. So the value of the flux can go anywhere between 0 and 1000. Okay.
So this character will be used for that.
B.
If we want to fix it at a particular point, I will say that it should not go below 0 and neither go above 0. It should stay at 0. You want to fix it at 0. We will use B. Okay.
Range.
Zero bound.
We are not giving any range. Right. So we have simulated, we have done FBA for this model with both conditions, aerobic conditions and anaerobic conditions. And what was the glucose uptake value? -10. Okay. So which is the first set of the codes up to here. Simulation for glucose under aerobic and simulation for glucose under anaerobic conditions. Okay. What was the objective function here? Biomass equation was the objective function. Okay. Can we do it similarly for the xylose?
Yes.
How we will do that?
Change reaction bound.
No, we will not change the objective. We are just changing the source, carbon source. Okay. We are just changing the carbon source. We will put the glucose uptake rate 0 because now no glucose is there available in the environment. Cell has to take another carbon source, and that carbon source we are providing as xylose. And we will switch on the xylose uptake rate. Okay, we will again update the xylose uptake rate as -10, which is we can see in your booklet. Okay, so we can give it a lower bound -10, or we can fix it at -10. Okay, we can do it either way. Okay. Also, if we want to do it in a aerobic condition, what we will give the lower bound of oxygen as -1000. And if we want to fix, if you want to do it in an anaerobic condition, we will fix the oxygen at 0, or we will give it a lower bound of 0. We can do it either way. Okay. So in this way, we will be simulating it under aerobic and anaerobic conditions. So try to do it on your system. Just you have to copy and paste. But before that, you have to re-update your model to the original condition. This thing you have to do it. Okay. So as you can see in your booklet, the first command is model is equals to model ORI. Okay. So you will change it to model_1 is equals to model_ORI, because model we are having for E. coli core. Okay. So do it as model_1 is equals to model ORI, or you can do it just for model also. You can just leave the E. coli core model here. I will also do it here for the online participants. I am updating my model as original model. Now, what I will do that I will change the reaction bounds. But when I am changing the bounds, I will have to take the model as model only, because now I have redefined our IGO 136 model as model. And the oxygen uptake rate I will define as 0 for -1000. First we will do for aerobic case. Okay. So then I will change the glucose uptake rate. And I will change the glucose uptake rate as 0. Okay. I have to check for the glucose uptake.
Yes,
I have to check for what is the
0.984.
0.984.
okay.
Value 1.
So this is the glucose uptake reaction. Are you guys able to follow?
This is 0.94
Yes.
values.
So now no glucose is available in the model. Now we have to provide xylose. So I will copy the xylose reaction.
इसका मान का मतलब सेट ग्लूकोज इनपुट फ्लक्स वैल्यू टू 0 फिर फ्लक्स वैल्यू हमने -10 पे सेट करी मैक्सिमम 100 पे फिर देखो कितना बना
Yeah. So this is the xylose. Just give me this thing.
ध्यान
0.982.
Okay. So no, this is wrong. I have to provide some xylose also. Na? So it should be -10. Cell has to be able to take some xylose. Okay. So I am providing 10 millimole per gram disulfide per hour of the xylose flux. Oxygen is
-1000.
have I? Yeah.
Semicolon.
No. Semicolons is there for so that if I put semicolon, the result will not be printed in the command window, but it will update. It will update. Yeah.
एक साथ
So
डाल
you're using model, not model_1.
Yeah, because I have redefined the original model as the IJ136.
आप पता
So I don't have to use model_1.
नहीं
I'm using model because it is written in the booklet as model. So the people who are not running it right now won't be having any confusion. Okay. So now we will try to maximize the growth. Okay. So what we will have to do as here we will have to use model only, and here we will define a new variable FV solution for xylose and aerobic. Okay. So we will be producing new solution every time. Yes. So FV solution for xylose aerobic is coming out as 0.8061. You can see that the growth rate was in the case of glucose, it was around 0.98. But in the case of xylose, it is coming around 0.80. Why? Because the biomass yield is a bit lower, because xylose, in the case of xylose, the biomass yield is a little less. So that's why the growth rate is also a bit less than the glucose. Okay. Now What we have to do? We will change the reaction bound of oxygen as 0. Okay. We have changed the reaction bound of oxygen as 0. So now we will do it for the anaerobic case. FB solution xylose for anaerobic and see what is the growth rate. So the growth rate is coming out as 0.16. What was the growth rate for glucose anaerobic case? It was around 0.24. So in the case of xylose, it is also a bit less. Right? All of you are able to follow? Are you able to do it on your
getting 0.98 to 4.
You are getting 0.98.
So, but in command instead of B, it is written B here, and you are writing 1. Because of that, the difference is coming.
Right, right. There is a mistake in the booklet. Okay. What is the mistake? Right. The oxygen uptake for the xylose under aerobic, I have given B. Okay.
Not written 1 here.
Where?
आपने इसमें 1 लिखा है, मतलब I had checked the commands here. You can see. But booklet में B लिखा है. L हाँ, lower bound. Technically,
yeah. So there is a printing mistake here. You have to just change. The first printing mistake is for the change reaction bound. In the second command, I have used the reaction identifier of glucose. So that is a mistake. Okay. You have to change it to the exchange reaction of xylose. Okay. So take it from the second command and paste it correctly into the first command. Are you able to get it?
Yet again, I'm getting 0.98.
Yeah, you are getting 0.98 because you have entered the reaction for glucose, the reaction name of glucose.
Xylose की जगह 1 ये नहीं आएगा.
Xylose की जगह भी I have also mentioned the lower bound is
This would be first one.
So copied it from here and change it to else.
इन्होंने जो लिखी है ना वो देख लीजिए, थोड़ा स्क्रॉल करना पड़ेगा.
Do one thing. You have model.ori here. Just define your model again as equal to model.ori. Just leave it now. Is equal to model underscore. Are you able to get it now? No. Just redefine your model as model underscore ori.
Okay. Now.
Yeah.
B is fine.
Okay. You have
It will be
yeah, you are
L,
getting this
lower
thing
limit
because you haven't
B fixed.
define you haven't हमें पहला क्या करना होगा, ग्लूकोज अपटेक वैल्यू को हमें 0 करना होगा. We have to define the glucose uptake value as 0. The first command. I was a bit confused, sorry. You have done it correctly, right? And then you have entered correctly the xylose uptake rate -10. Okay. So in spite of B, use L here. Okay. You have done it?
Yes.
Again, it is not working. And then change the oxygen uptake rate as L -1000 L. Is it right?
Not in model पर क्या है?
ऐसे ही डालना है L की जगह डाल के. ये इसका लैपटॉप थोड़ा स्क्रीन शॉट करके आओ.
वन है यार, ये L नहीं है.
उनका क्या मतलब है? L है ना, लोअर लिमिट.
L से भी नहीं है.
इसका देखना पड़ेगा, इसका नीचे करके
Change the glucose value. Yeah. Because yeah, you haven't changed that. You are taking the because it is defined in the model like this.
oxygen supply is 1000 for anaerobic.
वो B की जगह
Oxygen supply is anaerobic is 0. Oxygen input. Anaerobic is
So
0.
how many of you are able to get it? You are able to get it?
Again 0.98.
Again 0.98. Yeah. Are you able to get it now? No. Sure. You have loaded the model correctly? Okay. So the first command is glucose uptake rate type it 0. So this is not the glucose uptake rate as defined in the genome scale model. You should this is not the name of the reaction is different. This is the core model's reaction. Right?
Okay.
So how should I get that?
कौन से copy paste करना पड़ेगा now?
Go to the Excel sheet
I don't know. What is the answer?
and this is the reaction number. Go to this list. Search here for the glucose. I think search for glucose. Here reaction names. Search for the glucose. Find out.
Glucose ID.
Find out. Click on find out. Check for the glucose exchange. Yes, D glucose exchange. This one. Click on it. Copy this. So now you're able to get
बोलते हैं
the
ना
test
हमारे
value.
पास का है.
Are you able to do it on your system? Use it in single quotes.
अब इसमें जाओ.
So our online participants are able to understand what is going on here. How many participants?
Yeah.
Thank you.
Yeah, we are following it. Thanks.
You're not getting it.
I changed to model over model equals
So do one thing. Are you able to see the screen properly? See here. What are the commands that I will show all these commands in the
Find all
PowerPoint way. So first I have changed my glucose lower bound as 0.
Find all.
Are you able to see it?
Yeah.
You changed
0 L.
no. This is the correct command because the reaction of glucose uptake as defined as in the genome scale model is this one.
All of them as L. Lower bounds,
Yeah.
not the B's. The B.
You can use B or lower. You can do it either way. You can fix it as 0 or you can keep the lower bounds.
ये नाम क्या है इस पे क्लिक करो.
Yes, this is exchange underscore B.
फाइंड में यहाँ पे कहाँ
The reaction is different.
How many of you are still getting the error? You are able to do it? You are getting again error?
हाँ तो इसका नाम ये नाम कॉपी करो.
Go ahead with the
अब इसको राइट
with the xylose similarly. But the xylose,
अब करो एंड
or else you can go to the model also. From there you can.
अब नेक्स्ट
But you have to use model underscore 1 because you are
Or else
optimizing for model.
go to the
That's why I created the name of under. Yeah, this is aerobic. Now do it for anaerobic condition.
from
Oxygen uptake.
to have Excel, right? That whatever.
Yeah,
we
Excel.
can- From there also you can find
ठीक है बाद में दे रहा हूँ चल तो रहा है.
reaction list and find the. You can control F and
Excel.
yeah, this one you just copy paste.
यहाँ पे ये
We are not getting.
Not getting.
ये यहाँ तक तो कर लिया, नाम सर्च कर लिया था उसने।
यहाँ तक तो आ गया ये रिएक्शन करके चेंज बाउंड मॉडल है
Change the name.
ग्लूकोज एक्सचेंज
Change value.
Okay, glucose uptake rate is now 0. Okay, now change the xylose uptake rate as 10. You've done correctly, right? No, no,
गिविंग द वार्निंग
not in the model.
यहाँ
ना
पे कुछ दिक्कत है।
यहाँ पर आपने ये dash, you have used dash here. Just change it to underscore.
Okay, okay.
This reaction is not.
Correction is.
Use underscore here. This reaction is also not. Go to the Excel sheet of that model. Now search here in the reaction name in this column. Go into this column
यहाँ पे
ओके
and search for xylose.
Right, the aerobic, underscore aerobic, whatever.
Search for xylose here.
किसके साथ?
Aerobic and whatever. you-
Copy this name.
Tyler.
We can find the value here. Okay.
नीचे
Here.
Zero and zero, right? It must be anaerobic.
राइट हियर
So you can type the anaerobic. You can look at this xylose is again you are giving zero xylose. That means there is no xylose at all. Again, here it is zero. You are not giving anything at all. So make it 10 here.
Now run it.
Make it 10. Then
giving
xylose.
xylose.
Hi, xylose is a.
next है
That's
देन
auto import करना है।
दैट कॉपी दिस
there's some different
yes,
value, but you are doing.
Put all the constraints, but I'm not getting the value you want to.
You must be using some different model.
पुट है
To define the model you use.
First, like IJ.
X one.
This is the xylose aerobic.
0.16. Which one you are not getting?
0 and puts together.
Okay.
806. So this is the orbital.
Not at all.
change here from this
I have given.
so
Update that sheet. ये चेंज कर लेते हैं, इसमें गलत लिखा है ना ये। ये
तो तुम इसमें लिख लो
लिख लिया मैंने।
Aerobic.
Yeah.
check from
So now everyone is getting it.
yes
We are getting again error.
we will
You change the cell here.
देखा ना
This will be, make it L.
अब एनारोबिक के लिए जाएंगे हम।
Make it L. Enter. And whatever the was optimized.
ये जब optimize marks पे करें तभी use.
You can say
ये तो है ना।
The solution. What is the name of your model?
I think that's the answer.
Model
I said,
underscore one, right?
I want the first.
just change it to model.
एनारोबिक के लिए भी उतना ही आ रहा है
Type in model. No, delete it.
ऑक्सीजन एक्सचेंज की रिएक्शन देखनी पड़ेगी दोबारा से
Still coming point.
किसके लिए देख रहे हो?
Values to -10, -1000.
So
And
So 1000 minus 1000.
type in what is
You're not giving anything minus 1000 there. Oh, anaerobic to 0.
here you have put the
IG1366. Model is one IG1366. So now
बाद में भी जब
When you use this model,
इसको बंद करोगे तो
number,
इसमें define करेंगे.
because some
can
expression
do the same thing.
go ahead with that. Whatever there
When you are getting the yeah.
you're confused with the like the different model assignment. Like model 1 is equal to model 1. Model
Yeah, this model is the main model.
Oxygen uptake reaction 0.
क्या कहना
Model from big database.
Hmm.
Right.
This is the same reaction in the model in the IG1366.
एनारोबिक के लिए अपन
Check it again.
Model
change
change
ऑक्सीजन
reaction.
जीरो
Check it. Whether it is
ऑक्सीजन वाले का नाम।
I think some misalignment in the model. Whatever he is using, you're using. The misalignment is that's the reason you're.
जो अपन ने किया ना।
But when you follow this complete protocol, right? You will the same values. Or else what you can do, you can practice from
to remove the brackets
the same syntax. You will get
underscore.
download your own databases, right? You just copy paste the
Model
syntaxes and only thing you have to understand is the values.
yes
Aerobic and anaerobic and whatever the input carbons also you have to give xylose, glucose or something into glycerol. Sometimes people go for a glycerol and that you have to understand. So try to understand little bit logic and what.
Yeah, I used to complete that. One more
Maybe the modeling
identifiers.
misaligned.
The identifiers and
करना है ना अपन 1000 पे कर रहे हैं
I think it must be used like multiple model. And he shifted to some model.
What
That's
is
only the
glucose?
reason. Yeah. But once you start from the
Define the
starting
model
to end,
correctly.
you will get. Actually, here we have defined like one is that model underscore original and as well as 166 and again we come back to the model. That's the reason we are confusing. Just understand the logic, then later you can work.
In exchange rate की जो reaction थी,
Leave it here. Do one thing. I will type the
Once you go
see
home.
what I'm typing correctly.
identifier
Put in here.
जो ये जो identifier पहले वाली reaction
ठीक है ठीक है
ही करना है इसमें
So this was for the xylose simulation, and now we will move towards a simulation for product maximization. Okay, so most of you are getting errors because you are typing some different commands. That's why you are getting error. There is no problem. We'll get the correct answer. What I will do, I will retype all these commands correctly and then email it to you. Okay, then you run it on your system. You are having MATLAB. Okay, run it again freshly in a fresh manner and see what happens. Okay, yes ma'am. Okay, there is a question in the chat box. What is the question?
yes the question
Okay, so go to the next section, simulation for product maximization. So please focus on the screen and see you are typing the as I am typing here. Okay. So first thing first, we have to re-update our model to the original condition. Okay. So what is the command for that? Model is equals to
model underscore original
model underscore original. So type it correctly and you should see the result like that. Are you all able to do it? Okay.
model underscore original
If you run this command, raise your hands. Everyone is getting this answer?
yes
What is the model ID?
I is 01366
Okay, so now your model is now I01366. Okay. Now, right. Done?
Yes.
So the next thing that we will do, so when I was trying to maximize ethanol and I was setting the objective function as ethanol exchange. What we were seeing that the flux for ethanol exchange was coming out as 20 and the growth was 0. But this is not the practical case. We don't want that. What is the practical case? That if we are producing, if the cell is producing ethanol, it will be it will be growing, but it will be growing at a slower rate. Okay, so we will define the growth rate of this organism to a certain specific value, but lower than the maximum growth rate that is predicted. What is the maximum growth rate that was predicted? Was around 0.98 in case of the aerobic. Okay, and what was the anaerobic growth rate? It was around 0.24. So ethanol, generally the E. coli produces ethanol in anaerobic conditions. It doesn't produce ethanol in aerobic conditions. It always produces ethanol in anaerobic conditions because fermentation takes place in absence of oxygen. In presence of oxygen, no fermentation will occur because cell will use the TCA cycle and it will grow. So what we will do, we will define the growth rate. As you can see in your booklet, that take the growth rate as which is a solution in glucose anaerobic condition. Okay, so first we will do quickly find out the solution in the solution of FBA solution in glucose anaerobic condition. So model is defined as original model, and glucose is also, we will check what is the glucose uptake flux and how we can check that. Go to the model. and see the reactions. And was it at 185 glucose action reaction?
185.
Right? We can confirm from here. 185. It is the oxygen uptake reaction, not the glucose. Huh? 12 was the glucose uptake. Yeah, right. So 12 is the glucose uptake reaction. And now we will see what is the lower bound for the 12th reaction. It is -10. So glucose uptake reaction is fixed as at -10 as the lower bound. So we need not to fix it. Okay.
The lower, how did we check the lower bound for this? How did we check?
Yeah, no?
What
List
the
is
lower
there.
bound?
So in the earlier section, I told you a command, right? We could check the lower bound with the help of the command model.lb and put in the reaction ID. Just type in the command model.lb and in brackets type the reaction ID, which is 12 in this case. And it is answer comes out as -10. Okay, so this is how you can confirm. Okay. So now, since we want the answer of anaerobic condition, we have to, what we have to do? We have to define the lower bound of oxygen uptake as 0. So do it again. Okay. This we have to do. So now we will do the FBS solution. Okay. But here we have to put glucose in place of xylose. We are not providing xylose. Okay. Is everyone able to do that? We have just changed the oxygen uptake. These things. Model, change reaction bounds, model, and oxygen uptake value 0, and optimize CV model, maximize, just change the name of the variable as FD solution in glucose and anaerobic condition. Okay. Now, just maximize, press enter. Is coming out as 0.24. Can you see? Is it coming out as 0.24 for everyone? Raise your hands. How many of you are getting the correct answer? Now, okay. Now we will define a new variable and give it a name, growth rate. And we will make it equals to FBS solution underscore glucose underscore anaerobic dot F, and we will multiply it by 0.2. What is the meaning of this thing? That we are just taking 20% of the maximum growth rate in anaerobic condition. Okay. So we are fixing the growth rate equals to 20% of the maximum growth rate in anaerobic condition. Okay. So it is around 0.04.
Again, we will run that optimum.
No. We will run the optimum. We will run again the optimum solution, but we will have to change the. We will have to fix the biomass reaction to this value. Okay. You can see in the next command, model equals to change reaction bound model, and then the name of the biomass reaction is there, and the value that we are fixing there is equal to the growth rate. And then I have put B. B means fix it. Fix the biomass reaction at this value. Okay. And fix the glucose uptake rate is already fixed as -10, and fix the oxygen as at 0. And now in the next step, change the objective to ethanol exchange. Okay. So we are fixing the biomass reaction at a certain value, and we are changing the objective function as ethanol, and then we are optimizing the production of ethanol. And then we will see what is the maximum ethanol that can be produced with a certain growth. Okay.
So this is simply the multiplication of the F value.
Yeah, this is simply the multiplication. That take the F value of FBA solution anaerobic, okay, and multiply it with 0.2. Okay. So can you do it? Just copy and paste the next few codes. Fix the reaction. Change the reaction bounds. And which reaction bound you want to change for? The biomass reaction. Where is the biomass reaction?
Eight.
Go into the reaction, and this is the biomass reaction.
14 also. One.
Yeah, we can take any of them. We can take 14 or 19. Both are the biomass reaction. Okay. And what is the value that we want to fix here? We want to fix it to the equals to growth rate. Okay. And since we want to fix it at a certain value, we will use B here. Okay. And the next thing is glucose uptake value is fixed, right? We want to fix glucose uptake value. Okay. It is not fixed. So we will put the lower bound of glucose uptake. Here it is xylose. We have to put it here. Glucose. GLC. Yeah, -10. Don't take it from the booklet. It is wrong in the booklet. It is for the E.coli core model. Okay. And the next thing is put the lower bound of oxygen uptake 0. And now
glucose के लिए वो D नहीं है reaction में देखो एक बार.
no, it's right.
It's right.
It's right.
The lower bound.
So here we put -10 again.
Yeah.
The next
Fixing
second
करना,
reaction.
lower bound करना.
Lower bound.
Yeah. Lower.
-10. Okay.
You can check if you have typed the correct commands. I'm pasting it here.
अपन ने ऑक्सीजन को यहाँ पे कर दिया, अब हमने -10 कर
सब
दिया।
कुछ सेम, बस एल ही आएगा, एल आएगा, मतलब फिक्स नहीं करेंगे लोअर बाउंड की तरफ।
ग्लूकोज को कर दिया।
Check if you have the correct commands here.
So you glucose at underscore.
Can you see it correctly? These should be the commands. First, we have changed the reaction bounds of biomass reaction. Then we have changed the reaction bounds of glucose uptake rate as -10. Then we have changed the reaction bounds of oxygen uptake as 0. Okay, lower bound. Okay. Now we want to confirm that our objective function is correct. So we will use the command check objective. Model. So yeah, there were two biomass reactions in the model, a core one and a wild type one. So the objective function is set at the core reaction. So we have changed the lower bound of the correct biomass reaction. Okay.
When I'm typing the first, the growth rate is equal to solution, that part only is stuck. Just saying unable to resolve the need.
Okay. Have you done it correctly? Can you help her? Okay.
डिक्शनरी का प्रॉब्लम है, तो जाके जो
Able to resolve the meaning.
Answers are like very straightforward. I just need to
Okay.
change
So
तो
the workspace.
but this is where is correct or not. I don't know. Very low maintenance. Changing to external. This is fine.
Here is a generate here.
ये बीमारी मेरे
Not maximization. So I have done till this one. Okay.
Xylus one.
I think this is right.
But Xylus
Both are not
rates
तो मुझे इसको काम में करके दिखाओ, स्टाइल में कर रहे हैं।
how it
it won't this is not tough. And below that lower thing, nothing can happen. B means you're fixing both lower and upper at the same time. So the maximum thing that can happen is your lower limit or your constraint.
core model. The model IG1360. Core model का है. I was pasted these codes are. So don't just blindly follow it. Listen what I'm saying. हमें reactions भी IGO1360 use करते हैं. Next किसका है? So you can just run it this way. So you will be able to understand. हाँ ये करेक्ट. इसमें आपको ब्रैकेट लगाना है, अंडरस्कोर लगाना है. And in the case of glucose,
सारे पैरामीटर्स मर्ज के बाद से मुझे पहले हम तो नहीं देखते हैं कि इसमें शो कौन
then the correct option of taking.
पता है कितना
We'll get it. I'll give you. which because this is not set as the objective function. No? What is this? What are you trying to do? Here you are taking W wild type action. Use the core one. This. No, set it. Set the not. Now, now do the thing. अभी हमने ही किया ना
नहीं इसके बाद आपने कुछ किया है तो वही मुझे समझ नहीं आया क्या करना है
दिखाना जरा ऊपर करो उसको
just change the uptake rates of glucose to -10 and then uptake rate of oxygen to 0. Okay.
switch objective check.
After that, we have to change the objective. I run the command to check objective. Okay. I wanted to know if the objective of the model is wild type 1 or the core one. So it is showing correctly the core. Okay. So next I will change the objective function. I will redefine my model and I will change objective.
It's coming the same.
Model and comma, and I will change it to the ethanol exchange. I will find out what is the correct reaction name of the ethanol exchange from here. So this is the correct. So see, I will copy it from here and then paste it. Put it the single quotes. Okay. And then I will just close it. Now the objective of the model has been updated to ethanol exchange. Now I will run the command to optimize the objective function. And what I am optimizing for? Ethanol production. Solution I am trying to get for ethanol production. Yeah, simply. Yeah. So now the answer is coming out as 19.28.
With some biomass, it is producing.
Yeah, with some biomass. Okay. So this is how we do the product maximization simulations. Maybe you are not getting the right answer, but are you getting the concept?
Yes, sir.
Okay. What we are trying to do here. The answer you are not getting because some of us are getting confused because of the wrong commands. Okay, because of the wrong reaction names or the wrong model that we are using. But you should get a concept. You are getting the correct answer. Okay. So similarly, as you can see that we can maximize for ethanol, we can maximize for lactate, for succinate, and for the other products also. We have to just use the. We have to just change the reaction objective. We have to update the objective function to the correct to the different reaction and then run the same command, optimize CB model and maximize that reaction. It will try to maximize the product. Okay. So next thing is how do we simulate for a reaction that is not present in the model? So we have to, for example, butanol is not produced by E. coli into the native state. We have to add these reactions. We have to add that pathway into the E. coli model, and then we will see how it can produce or not the butanol. So for that, come to page number 29 and just clear your command window. And what we can do that, just clear your workspace also, so that clear your work. All of you clear your workspace by typing in clear. Okay. Now again, type in the command model underscore ORI equals to read CB model. Type in the file name IJO1366.xml. Close the quotes and press enter. It will take some time to load the model. Already 5:30. It will take around half an hour still. Okay, so be patient.
एरर दिखा रहा है.
So meanwhile, it gets loaded. Just check it out, the codes that are there on the page number 29. Get some idea what the code want to say. So first thing is that it is defining the model equals to model original. We want to keep our original model file intact, and we will be changing the model, only the model variable. Then we are taking the growth rate as 80% of the anaerobic growth rate in glucose. Okay. Then we are changing the lower bound. We are fixing the biomass reaction at the growth rate equals to growth rate, and then we are changing the bounds of glucose uptake. We are fixing the glucose uptake and fixing the oxygen uptake. Yeah.
What formula?
Everyone has loaded the model into there. Okay. So type in the next command and define another variable as model equals to model underscore ORI. Okay.
No, not it. Right.
CLC. Now, yeah, ma'am. Now can you again do the same thing? Find out what is the solution for glucose in anaerobic condition. Okay. We will calculate again the FB solution glucose in anaerobic condition. Okay. So how we will do that? Glucose uptake rate is fixed already in the model by default -10. Okay. We have to just change the oxygen lower bound of the oxygen uptake rate. Okay. So we will change it. Model equals to change. We will just use the previously typed commands. Yeah. Model change reaction bounds, oxygen uptake, and we will fix it at 0. Okay. And then we will do the optimize the model. And we will name it as FB solution glucose anaerobic. Okay, ma'am, are you able to do? Everyone should be able to do now. Okay. So the answer should be coming out as 0.24. Is it coming out? Change the reaction bound for oxygen, ma'am. Fix the lower bound at 0 for the glucose uptake. Your reaction name of the oxygen uptake is different. Type in the correct reaction name from the Excel sheet. From the Excel sheet, type in the correct name. The correct name is this.
ये वाला इसमें कौन सा करा है?
ऐड करेंगे अपन इसमें।
नहीं मतलब अभी इसमें है ही नहीं।
इसमें नहीं है ना ये अभी अपन किसी डेटा अपलोड किया है।
This should be the reaction. This one. Check it correctly.
What are you doing?
Is everyone able to do it? Well, okay. So, ma'am, now you are getting correct answer.
1.15.
So now we have the okay. Now we will define another variable as growth rate. We'll define another variable as growth rate,
Another big condition, right?
which is equals to
The variable.
FBA solution
Molecular change reaction bounds.
underscore glucose underscore
Butanol addition.
anaerobic,
Yeah, but we did first this one.
and we are taking
And
the
we
objective function of this solution.
did this one.
Okay, so we will put dot f,
Right.
then we will multiply it with 0.8. Okay. So we are taking the growth rate as 0.19. Okay, we are only taking slightly low value than the maximum value.
Why we multiply by 0.8?
So because we want to take the 80% of the maximum
20
growth rate in anaerobic condition. Okay. Now the second step is the third step is fixing this value of growth rate to the biomass reaction. Okay, fix the biomass reaction to this value. So can we do that? Change reaction bound. Yeah. Yeah. Use this command. You have used it previously. Model equals to change reaction bounds. Model underscore biomass reaction name, comma, growth rate, comma, B in single quotes. Okay, I am pasting this reaction in PowerPoint. Please make sure you are typing it correctly. Model change reaction bounds, model, comma, biomass EC underscore IGO 136 underscore core underscore 53 P95M, single quotes closed, comma, growth rate, comma, B in single quotes. Is it same? Okay. Everyone. Yeah. Next thing is on it. Okay, we have fixed the biomass reaction. Glucose uptake reaction is already fixed. Okay, and we have also fixed the lower bound of oxygen as 0 as we have calculated, right? So now our model is it is ready to add the butanol pathway. So the first reaction that we want to add in the model is make acetoacetyl CoA from 2 acetyl CoAs plus release 1 CoA. This is the first reaction of the butanol production. From where we have taken these reactions? We have taken these reactions from the KEGG pathway. If you search for the KEGG pathway, just type in the Here is the here. Butanol pathway, क्या है? So you will be getting this thing and see it in the full form. Yeah. So here we have butanol. Okay. So we will reverse, we will go in reverse direction. So butanol is being produced from butanol, which is coming from the butanoyl CoA, which is coming from protanoyl CoA, and which is coming from 3-hydroxy butanol CoA. Okay, and the 3-hydroxy butanol CoA is coming from the acetoacetyl CoA, and acetoacetyl CoA is coming from the acetyl CoA. So this pathway we have taken from the CAC database only, which is an authenticate source of reactions. And this pathway is present in the Clostridium what is the name of that organism? You can check it here. Yeah, Clostridium acetobutylicum. Okay, so we have taken these reactions from this pathway, and we will try to paste it correctly. Okay. So where is my code? Right. Take this first reaction, Control C, do it on your system, same thing. We have defined but1 a variable, and what is the variable but1? It is the reaction. Okay, and this is the reaction which is defined as produce one acetoacetyl CoA from the two acetyl CoAs and release one CoA. Similarly, I will define the second variable, and the second variable is producing 3-hydroxybutyryl CoA from the acetyl CoA, and with the help of one NADH, it is reducing. It is reducing the acetoacetyl CoA. So second variable has been defined. Are you able to follow?
Yes.
Now I will define the third reaction as but3. Please do it also on your system. The but4 reaction is defined as production of butyryl CoA from the protanoyl CoA. Next, we will produce butanol.
Sorry, I want to.
Yeah.
This the concept of addition of butanol pathway reactions from the CAT pathway. Can you please explain that concept? I didn't really get.
Okay, I'll explain. Can you just please do this thing?
I'm following.
You're following it, right?
Yeah.
Let me add just one reaction, then I will tell. So this reaction is producing one butanol 6 reaction. I have to add a last reaction of butanol exchange. Why? Because I want butanol into my extracellular medium. So I want this butanol. Yeah, this but7 reaction will secrete the butanol from cytoplasm into the extracellular space. Okay. Yeah. So to answer your question, we don't have butanol natively produced into the E. coli, right? So butanol is produced by this organism called as Clostridium acetobutylicum. Okay. So I have taken these reactions. I have taken these 6 reactions which produce butanol from acetyl-CoA in 6 steps. From first step is produce acetoacetyl-CoA from acetyl-CoA, then from acetyl-CoA produce protanol-CoA, then protanol-CoA to butanol-CoA, butanol-CoA to butanol, butanol, then butanol. Okay, so we are defining all these reactions. In actual conditions, we have to put genes of these reactions, genes which are responsible for these reactions that are taking place into the Clostridium. And then we have to clone these genes one by one into E. coli. Okay, you will be doing it while we will be demonstrating into the wet lab conditions. The same genes, the same reactions we will be adding into the E. coli actually. Okay, using the CRISPR-Cas9 technology. Okay, the same thing we are doing it here on computer. You will be doing it in the E. coli actually. The same pathway will be integrated. Okay, now you get it.
Yeah.
Any doubts?
The reaction which are written in this format, how do we get this?
Yeah, that's the question. How do you get the reaction?
How to get this section? Okay, so yeah, good question. I'll tell you.
So now each of those, they are metabolites, right?
These are metabolites, yeah.
And the metabolites, of course, because of various genes, of course, affect a particular metabolite.
Right.
So is it that the multiplex of those genes, or like how you identify, within this particular metabolite, let's say there are 10 genes.
Mm-hmm.
So how do we know, are we to do with all the 10 genes, or how do we, I don't know. Yeah, how?
No. First of thing, metabolite is not because of a gene. Gene produces enzyme.
Yes.
Okay, not the metabolite.
Yes.
Metabolite is produced through reactions.
Yes.
Okay, that will be used up or produced into a reaction.
Now, talking
it is not directly related to the metabolite. Gene is not directly related to it.
I know it's not directly related.
Right.
I'm only talking in context of the gene metabolite general reaction.
Right.
Relationship.
Right.
So of course, the genes, of course, there are just various pathways and all of that produce metabolites. So my question is, those are metabolic pathways.
Right.
You're picking those rates from. So how do you come about the genes? That's my question.
How do you come about the genes? Because we know the metabolic network of Clostridium acetobutylicum. What is the name? I can. Yeah, Clostridium. I will say for simplicity. So we know the pathway in Clostridium. Okay, we know the genes that are responsible for these reactions in Clostridium. So this is our hypothesis that if we clone those genes into the E. coli, because E. coli can produce acetyl-CoA from glucose, we just have to go from acetyl-CoA to butanol. Are you getting my point?
Yeah.
Are you getting?
Yeah.
So how do we get from acetyl-CoA to butanol? Through these reactions, right? Two acetyl-CoAs can produce acetoacetyl-CoA. Then this acetoacetyl-CoA can be used by this enzyme and to produce 3-hydroxybutyl-CoA. Are you getting it?
Yeah. So how do you now get the reactions like this?
Yes, the reaction that.
How do you get these reactions? How do you add these reactions? Yeah, I'll tell you.
I have one question. From the KEGG pathway.
From the KEGG pathway only, yeah. Right? You can see that in the successive steps in the booklet, when I'm changing the model is equals to add reaction, model there is an identifier of reaction R00238, comma but1. What it is saying, this reaction identifier, if you search this reaction on Google, it will show you the reaction in the KEGG pathway. Okay, just type in this reaction. I will type it for you. Go to the Google. Type in R00238 and type in KEGG here. KEGG reaction R00238. Get this reaction, right? 2-acetyl-CoA is equals to CoA plus acetoacetyl-CoA. Okay, so this is the authenticate database of reactions. We can get our reactions from here.
So in the MATLAB code that you have written, like these are the identifiers for
Right.
that particular reaction.
Right.
Is there any set of rules or convention through which we have to like go through by those rules?
Obviously.
That we have to mention that.
Obviously.
Or is it like everyone makes the model, makes their different set of identifiers? Is it so, or is it a defined set of rules?
No. In the case of a particular model, people can use different identifiers. People can use different identifiers.
Have to go through all the identifiers.
But there are some modules in the model that KEGG IDs.
Okay.
The different attributes for KEGG IDs, the metabolite IDs, they have to put in the KEGG IDs or the metabolite IDs correctly in those models.
Okay, so can we, like, instead of putting those identifiers, we can put the KEGG ID into this?
No. That will be difficult, right? KEGG ID is, this is the KEGG ID, R00.
Okay, but those identifiers may be different for different models, no? So that...
yeah, identifiers can be different for different models, right?
Okay, so I'll have to check all of the IDs into that model firstly, and then go into that.
Right.
And if suppose we are trying to use the KEGG IDs into that commands, so is it possible to like run these commands with the KEGG IDs?
I'm not getting it. What you want to say?
The identifiers which we are defining, the reactions, like if we just denote the terms, the KEGG IDs, I mean, like instead of the identifiers, we just define the KEGG IDs and we just recall them or call them in the commands. So is it possible?
It will work, okay? I'm doing it the same way, right?
Okay.
I'm doing it the same way. See. Now the next step is we have defined the variables. Now we will add them to the model. We will change the model. So the first, I will copy them one by one so that you can understand. So I have added the first reaction with the identifier R00238. Okay. Second reaction. Now I will go all of them at once. Now, just check our model and go to the reactions part. Okay, and see that 7 reactions have been added. R00238, R01975, R03096, and the last reaction is butanol exchange. Okay, so now you are understanding what is happening here.
Reactions, how are you detecting?
So now our E. coli is ready to produce butanol. It can now produce the butanol.
Copying it.
Yeah, any questions?
I'm still a little confused, like when you are saying this R identifier is there. Right now, I don't know what all genes will be, but with the KEGG pathway, I can follow the arrows. From there, how do I find out these values? जैसे आपने कहा ना कि इसको put it in Google, मुझे तो पता ही नहीं है ये है तो मैं Google पे कैसे डालूं? तो I have to see from the KEGG pathway.
Yeah,
That I want to know how.
Okay. So you can see here that when I go to the, here.
ये पूछने उन्होंने ये बताया कौन से बच्चा
No, slide. This is nothing. Identify कैसे करें? Count the reaction when we are trying to produce. Yeah, produce this one. 6 reactions. If we are adding extra.
So, ma'am, the first thing is that we have to get the net metabolic reactions. Okay, we have to have the idea that that reaction is present somewhere, right? If we don't know that this kind of reaction actually takes place somewhere in some organism, okay, then we cannot add it, because we have the idea that yes, Clostridium produces butanol with the help of this reaction. So we can go to the metabolic network of Clostridium, and there we can find it out. Okay, here it is not showing, but if I type in that, so for example, if I type in Crotonil CoA, Crotonil CoA Scale. Okay. So it comes out, and here it is showing that it is present in the butanoid metabolism. Okay. Here, and these are all the reactions that it is involved in. So we can check these reactions one by one, and that's how we can search for the reaction. Okay, it is a bit time-taking process, but it is done that way.
So individually, we have to be.
You have to look for that, yeah, which reaction it is. Okay.
I mean, from the cat pathway now.
Yeah.
Those numbers, those specific reactions, we have to open it in area like that.
Right. If you simply know the reaction, for example, Protonol CoA 2, what it is producing? Butanol CoA, yeah, butyl CoA. Okay. And type
We
in
we can find the butanol also.
Yeah.
The same way.
So hello. Yes, sir. हाँ सर अभी चल रहा है तो थोड़ा अभी आधा घंटा लगेगा ऐसे. Yes. Yeah, we are about to wind up. Yeah. Yes, sir. है.
Yeah.
Okay, so
We had 7 reactions here, yeah.
yeah.
And I could find 5 reactions in the data CAG database.
So first reaction is acetoacetyl CoA, right? That, and the last reaction is the exchange reaction that we add by our intuition, by our knowledge that we want the butanol out of the system. Okay, so that cannot be found in there because yeah. Why should we should include every single compound in the vector of the metabolites too? So when we use the reaction when we will be using this command add reaction, it will automatically add the metabolite in the list of the compounds. Okay. Someone is asking in the online question that, yeah, okay, we have to add these metabolites also, because these metabolites are not naturally found in the E. coli. So since we are, here we have defined these reactions, and this butyl-CoA, protanol-CoA, these are not naturally found. But when we type in this command of add reactions, it will automatically add these metabolites. Okay, so we don't have to worry about it. Yeah. So next thing is, what is the next thing? We have to change the objective to the butanol reaction, butanol exchange reaction. So it's good that people are following online also.
Informative.
Change objective. Right. But for in place of ethanol, I have to put butanol. BUTOH. And we have to remove E.
Change objective.
And, correct. And now I will optimize CV model. Okay, I will name it as I'm using glucose. I'm using doing it in anaerobic, and I am optimizing butanol. BUTH. Okay, let's see what is the answer. So the answer is coming out as 8.5. So this is the maximum flux that can be generated. Okay. So this is the maximum flux. All of the flux I am diverting towards the butanol production. What is the growth rate? That is, can you check the growth rate here? Check it yourself. Yeah. What is the growth rate here?
Biomass reaction.
Growth rate.
ये नहीं तो मॉडल बनाना
14th or 15th, 19th. We can check any of it. It should be same. 0.19. Okay.
.1932.
Right? It is 0.1932. Why it is not coming here? Okay, it is not using it as a growth rate. Wild type is not used. Okay. Why it is 0.19? Okay, tell me. Because we have fixed it. Initially, we have fixed it at the 0.19. That's why it is keeping it at 0.19. Okay. Okay. So this part is done up to here. Come to the page number 30. So now we know that our model can produce butanol. It has the potential to produce butanol. Now we have to identify what are the targets, what are the genes that we need to knock out or we need to overexpress so that we can produce the butanol. Okay, so how can we do that? How can we identify it? We need to see that while we maximize the butanol production, what are the other fluxes, how the other fluxes are behaving. Okay, So the fluxes which are also increasing while we are increasing the butanol, those fluxes are basically, it means that they are necessary for the butanol production. Can you understand it? And the fluxes which are decreasing, it means that those fluxes, those reactions need to be shut down. Yeah, they have to be deleted kind of for the butanol production. So we have to identify which reactions or which fluxes are changing significantly while we are trying to maximize butanol production. Okay, so how we will do that? We will try to increase the butanol production slowly from 0 to the maximum value, that is 8.5 in small steps. Okay, so and then we will see how the other fluxes are behaving for each value of the butanol. Okay. So yeah. So this process is called as product scanning. We are scanning the whole of the fluxes while we are trying to maximize the butanol. So this process is called as product scanning, and it helps us to identify the suitable targets. So again, we have to define right now what is our objective function. It is butanol maximization, right? We will again reset it to the biomass reaction. So we will define a biomass reaction. We will change the objective function. I will copy this name, Control C. This is the name of our biomass reaction, right? Okay, and I will define a function. I will define a variable, biomass reaction. You can see it in your booklet. Biomass reaction name. And I will paste it here. Okay. Why I'm doing it? Because I don't have to type again this difficult name again. See, I cannot update again the model as original model, because if I update again, all the changes I have done, I have added 7 reactions into the model, they will be reverted back. Okay, that thing I don't want. So I will just keep on changing the updated model only, the old model only. And yeah, I will change the bounds of this reaction. Change reaction bounds. Yeah. And yeah, Control V. And here is biomass reaction name. And I want to keep the lower bound as 0. The lower bound can be 0. And something wrong happened. Yeah, comma is missing. And I want the upper bound as 1000. So why the biomass reaction, the lower bound for biomass reaction, can it be 0 or can it be negative? Because either the organism will grow or it will not grow. It cannot go negative side. Okay, it will not disappear. Yeah. So the upper bound is again 1000. So by default, the lower bound for the biomass reaction is 0 and upper bound is 1000. Okay, it takes any value from 0 to 1000. Okay, so next thing is I want to change the reaction bounds for glucose uptake rate as -10. I think it is already fixed at -10. I haven't touched the glucose uptake rate. Okay, I don't have to change it. So the next reaction is change the objective to the biomass reaction. Yeah, that thing I touched. I need to re-update it because I changed the objective from biomass to the butanol maximization. So I will update it. I will use the change objective, and here I will again the biomass reaction. Okay. Now this part is simple as we have done it again many times. Now focus on this part, fluxes. I am defining a new list of fluxes, and it is an empty list. Okay, these square brackets mean that I am telling MATLAB that take a variable and make it equal to an empty list. Okay. Now what I want in this empty list, I want to add the fluxes for each simulation. I will be running the simulation many times using a loop. I am using here for loop. And how I will be using for loop? What are the conditions I am defining? That take for I equals to 0 and increase at 0.1. First value take I equals to 0, then take next value 0.1 and go up to the maximum value of FBA solution, this one. What was the FBA solution for butanol maximization? 8.5, right? Someone quoted it correctly. So what was the variable name? I forgot it. I have to copy and paste it. Change objective. Yeah, this was the name of the variable. Control C. Control V. Okay. And take its flux value, which is .f. Okay. Next. And for these values of I, change the bounds. Every time it will change the bound. You can just copy and paste also. Change the bounds. I can also copy paste. But,
लास्ट में भी एक कॉलन
R03544 I, and I'm using B here. What is this reaction R03544? Can you see it from the previous page? Check it. What is this? Sixth reaction, right?
Library name.
Yeah.
Optimize CV model. Take the updated model and maximize. आधा घंटा तो लग जाएगा आराम से. And then end. Okay, so the loop starts from here for I equals to 0. And what is the step we are taking? 0.1. Increase the I from 0 in step by step 0.1. Yeah, see.
Why are we taking R035?
Yeah, R because we want to maximize this reaction, right? In R03544, butanol is being produced. Okay, so in this reaction, we want to maximize this reaction. We want that our butanol increases stepwise and reaches to a maximum value of 8.5. So this is the final reaction. Okay, we want to change the flux of this reaction, right?
Because okay, the maximum flux is 8.5.
Because the maximum flux is 8.5. So we are fixing every time the flux of butanol production and increasing it bit by bit. Okay, and then see, and now we will maximize growth only. Okay, we have changed the objective function to the biomass reaction. Okay, we are not maximizing butanol here, but what we are doing, we are forcing the E. coli to produce butanol plus simultaneously grow. Okay, grow while producing butanol to a certain value. Okay, but do it in a stepwise manner. Why we are doing it in a stepwise manner? So that we can see what are the other fluxes which change. Okay, we will see it in a short time.
So we are going to start increasing it from 0.
From 0. Okay, you will see that its growth rate will also decrease as the butanol is maximized. The growth rate will also decrease. Okay. So then I have put the end here. Yeah. And all those fluxes. Yeah. Right, right. The third line I forgot to enter. Thanks. So the third line says that every time you calculate these fluxes, okay. Copy paste करने से बड़ी हो रही है.
Did you done it? Something else. Done for the day. I want to leave. Planning to go somewhere outside. Fine.
Yeah, I know. We are not getting every time we getting error from the last.
No. It's the error is coming because typing mistakes.
So it's very important.
Define new, just try to resolve it.
Every product scanning, and the last is
ये रिएक्शन सही आ रही है ना, उसके बाद वाली में गड़बड़ हो रहा है. biomass reaction name equal to C.
Fluxes, update the flux list, fluxes list. Okay, so this last command tells that every time you calculate the fluxes, add those fluxes into this empty list, okay, and then re-update that flux rates into the next column. So every time it will calculate the fluxes, it will add it to the list. Then it will calculate again, add it to the list. It will do it how many times? Till it reaches the maximum value of the flux, okay? Now I will put the end. Right. So it is calculating, unable to resolve the name FBA product scanning.x. Where is FBA product scanning.x? Okay, right. Okay. So what is the thing? These commands were generated for an older version of MATLAB where the answer was solution of fluxes was coming out as X, but here the solution vector is coming out as B. That's why it was showing error.
No.
Okay, it is not still doing it.
No, actually, there is no underscore there.
Huh?
This in the third line. If,
Okay, the
if be a product scanning is no underscore is there.
no underscore is there, correct. Okay. Yeah. Thank you. Here,
Yeah,
The second line LP,
FB product scan.
you can give underscore there.
So now it is
not B नहीं है,
Not
dot X is
Dot X. I've done this. Sign of that.
dot V.
No, dot X. Dot X.
Where is the dot X here?
V है ना, V.
Yeah. So solution comes out as V only, right? Yeah.
Dot X.
ठीक है.
Example here. These are the fluxes. So the V is correct.
But इसमें भी underscore नहीं है.
अंडरस्कोर की जरूरत नहीं है. एंड इतना भी काम करता है कि नहीं? Yeah, it's working. Okay. So it happens many times while you write a code, you have to change it. So yeah, it has calculated and see what is there in the fluxes. See, are you able to run these commands on your system?
No.
No. Why?
Getting error.
The result.
एरर आ रहा है ना सब.
Error आ रहे हैं.
हम लोग देख ले रहे हैं, ऐसे आप कर रहे हैं, हम लोग देख रहे हैं, उसके बाद ये वीडियो से करके फिर
नहीं
आगे
अभी तक का तो सारा हो गया, यही वाला
लास्ट वाली जो रिएक्शन.
Yeah, it was a bit tricky, I understand. So it has calculated fluxes for 86 times. Okay, in each step it calculated fluxes and it added those calculated fluxes to the fluxes list. Okay, now what we will do, we will copy all these fluxes. Control A, Control C, and I will make another sheet here and paste it. Okay. No. I will do it here. Somewhere here.
Achha, if this is giving like 86 number of fluxes, so we'll have to like validate it with 86 set of experiments for this.
No. You don't have to validate it. This is we are doing because we want to see how the fluxes are changing from 0 to 8.5. How the other fluxes are behaving. Okay, because I could, in order to see that what changes are happening when the butanol is being synthesized, I have to see how the other different reactions are behaving. So I'll show you how. So these are the answers of these are the flux values. Okay, I don't know which reactions are those. So I have to copy and paste the reaction names also. So the reaction names were here, right? These are the same reactions, but these reactions are not having the butanol exchange reactions. So I have to take those reactions from here on. There is a model. Yeah, model reactions. Control C. And yeah, I will add it here. Okay. So you can see that here, this is the biomass reaction, and see how the biomass reaction is changing. Initially it is 0.24 because it is taking the maximum value of the anaerobic growth rate, and it should start decreasing. 0.23, 0.22, 0.21, and finally it comes at 0.19. The lowest value of biomass reaction is 0.19. So this is the maximum growth we are achieving while achieving simultaneously the maximum butanol production. We can see what is the maximum value of butanol production here. It is 8.5. You can see this is the butanol exchange reaction. Okay. Are you able to see it in the? Can you see now?
Yes.
Yes, it is visible.
So, right.
Wait.
8.5. Okay. So, how we will see that how the other reactions are behaving. See, most of the reactions are here 0. Only some reactions are changing significantly, and we want those reactions only to change very significantly. So since I have done this exercise previously, I know from my previous knowledge that these reactions will be changed. I will be showing them because it will take a lot of time to search for those reactions. Okay, because I have done this exercise previously. I know that the pyruvate dehydrogenase reaction I have to search for. Yeah, this is here. So you can see that initially the pyruvate dehydrogenase is 0, and it starts to increase significantly. It reaches to a maximum value of 17. Okay, you can do it in other way, like for example, how we can do it. This is CM. If I and this is CL. Okay, so if I put in here CL, sorry, I click this cell and minus this cell. Okay. What was the name of that cell? A1.
E.
Huh?
E1.
A1, right?
E1. Elephant.
It should be E1. It should be A1, right?
You have pasted in the 4.
Okay, fourth cell I posted. And then it will calculate the difference. So no difference because this reaction doesn't changes at all. It is not having any flux. And if I calculate the difference for every reaction,
कैसे लग रहा है? तो लेकिन वो लाइव कैसे पता नहीं पहले ही सिर्फ खुद में लैपटॉप.
okay, so through this thing we can see which reactions have changed very significantly. And if we divide this value, no, not in that way. So leave this thing aside.
आगे लगी. So the maximum changes will be based on the first reaction into the last, the difference between the initial and the last.
Yeah, the maximum changes.
Okay.
Yeah, I calculate the difference between them.
We
right?
can make put a filter and then we can set maximum to the lower in the CM column. It will come all the reactions.
Yeah, we can print that which reactions have changed significantly, the fluxes which are greater than a certain value, and we can select those values, right? So it will take a bit time. So I will just show you that PDH, as I showed earlier, that PDH is increasing. Okay, the flux of PDH is increasing, and the flux in PFL, the next flux is PFL. Okay. PFL. Okay, so the flux in this reaction decreases very significantly. And it comes down as 0.74 from 17. So from this exercise, we will know how which reactions are the targets of our genetic engineering. So we find it out that PDH needs to be overexpressed and PFL needs to be knocked out. Okay. So now we will do that in our next steps. We will try to change the bounds of these two reactions. Okay. And see that how the model behaves. We'll again go to the MATLAB. Close it out. So we will fix the growth rate. To be equal to, it was already fixed, right? No. So initially what we were doing, we were trying to maximize the production of butanol. But as you can see here that in the solution, where is the solution part? Yeah. So when we try to maximize the butanol production, we also get some absurd values of fluxes also. For example, some fluxes are coming very high and some fluxes are coming very low. As you can see that this is -1000 and 1000. So you will get many of such values like that. So it happens because the way the solver solves the linear programming problem in case of maximization of the objective function, it happens in the case of maximization, this problem comes out that many absurd flux values will be coming out. Okay. So the other way of solving this linear problem is by trying to minimize the total fluxes. And if we try to minimize the total fluxes, relatively the answers of the reaction, they are more reliable values. They will not, these absurd values which are coming out, they will be reduced. So as you can see here, FBA solution min, I have used the function FBA solution min optimized CV model, and then in place of max, we have used min function and taken 1 as the next parameter. So the optimization problem can be solved in 2 ways, either trying to maximize the objective function or we will try to minimize the total fluxes. Total fluxes means all of the reactions. Okay. So in this way, what is the benefit of using minimization? We will not get the absurd fluxes. Okay. So just try to add these codes and we'll try to run them. Okay. Taken from the data.
I mean, okay.
Unable to resolve the name solution glucose and aerobic because yeah.
तो फिर जाके
The reaction is not in that.
The same error I was getting that time.
Reactions. Okay, here the same reactions. Right. This reaction is not present. So we will run each line one by one to check for it run properly. Yeah, no, this is not there in the model. Now it's run. Okay. Next is this. Change the. We don't have to change it as it is fixed. Oxygen is also fixed. We have to change the objective. No, we don't have to change the objective function as it is also fixed. And we have to just calculate the FBA solution minimization. Okay, so it is it's showing anything? No. No solution is calculated by this solver. Okay, objective has been changed or not properly.
The name is different. Biomass underscore EC. Name is different. There is no EC underscore.
Right. I have to retype it. This I had run already, right?
You have to give the just biomass. There is no EC.
Yeah, so where I am using EC? This EC.
Sorry, whatever the next one you run, I think that
EC is there, right?
no, scroll down.
Okay. Here it is correct. Change direction bounds and change of objective. Okay. Right. Here I have to change the correct biomass selection. And now okay, I will type all the code again. I fixed the growth rate. Change the objective. Oxygen 0. यहाँ तक we have done it correctly. Okay. From here, just showing some problem. Why? Because I have run these codes incorrectly. Right?
What you can do,
Right.
what you can do if you have a final interpretation, just give the just like an overview and the rest of the coding part, type it and send it to everyone. Now you can wind up.
Yeah, it's getting a bit late also. So we are having some problem here, and we can do one thing also. We can have a 1 hour session in some other day of the workshop. Okay.
Last day or
Maybe a last day or
whenever.
Yeah.
So what do you do before that? You just write the code and send it to
Yeah,
everybody. Let them have a
let them run
exercise.
Yeah.
And now if you want to tell the overview, just give the overview.
Yeah. So this problem is coming out because of the error in the code. Maybe it's written incorrectly, but the concept is that we identify the targets correctly as PDH, pyruvate dehydrogenase, to be overexpressed, and PFL, the pyruvate formate lyase, to be deleted. You had seen it in that it was decreasing. So what we need to do that we need to fix the value of PFL to be at 0, and we need to fix the upper bound or the PDH value to be at the maximum value which it was producing, that is 18. You can see here that I have fixed the upper bound of the PDH as 18 and lower bound of the PDH as 10. Okay. So it is because initially the PDH value was 0, but we have taken here as the upper value as lower value as 10. So we want a minimum expression of PDH, minimum overexpression of PDH. Okay. So and we have fixed the value of PFL as 0, and then we try to optimize the model. Okay, this is the FBA solution 1. Okay. So it will give a certain value of butanol, but if we further want to increase the titers of butanol, we can also delete some obvious targets. For example, as we have seen earlier also, that butanol was being produced from the acetyl-CoA. Okay, so if you go to the core, the central carbon metabolism of the E. coli, you can see that the acetyl-CoA is used for the production of formate with the help of, okay. So formate is, in the production of ethanol. So if we stop the production of ethanol, the acetyl-CoA will be redirected towards the production of butanol. Also, the next gene that can also be deleted is lactate, because lactate is also produced with the help of acetyl-CoA. So the next obvious target is lactate. So if you run those codes correctly, so the titers of butanol will be increased again after deleting these 2 genes also. Okay. The problem that is happening here is because I will tell you, these codes were run on a different version of MATLAB, but for your convenience, we decided to install the latest version. This version of MATLAB is the latest one, and therefore this some problems are occurring here. Okay, so what I will do? I will try to update this code for the latest version of MATLAB, and I will send it to you, and also to our online participants, and we'll see what are the results coming. Okay, and we will try to have another 1 hour session with you. Okay, and see if you are able to run these codes properly. So thank you for your patience. This session was a bit extended, and I thank you to please stay here and listen whatever we are doing. I know it is difficult because all this exercise is very new to all of you. Okay? And I thank my online participants also. Be patient, and yeah, we'll try to inform you that when we will have this session, remaining part of session we will cover definitely, and with the updated code, and we will be sending the updated code to the online participants also, so that they can run this course. I'll try to say to the online participants that meanwhile they can download and install the trial version of MATLAB into their systems, and they try to install the COBRA Toolbox also into their system, so that they can run the COBRA. Yeah, I will provide these COBRA commands. First, I need to change these commands so that it can run properly into the new version of MATLAB with the COBRA. I'll definitely do that. Yeah. Thank you. We will close this session right now. Thank you to all the participants, and we will soon update you about this session, the remaining part of this session, and I will update you about the codes also. Meanwhile, try to install MATLAB and COBRA Toolbox. Try to install the trial version. I will send how to install. I will update you how to install the MATLAB and how to install the COBRA Toolbox into the MATLAB. Thank you. Thank you very much. Yes, sir, almost.
Can you help with